With pride and pleasure, I welcome you all to the final session of this SPSS workshop. So as we all have experienced for last 2 days, Sharma sir is explaining everything with utmost simplicity and with appropriate examples. So without a further delay, I request sir to please continue with the session. Sir, please continue.
Okay, thank you, Dr. Sangeeta, Dr. Rajesh, Dr. Shweta. Today is the last day of this workshop, and I think there are many messages, there are many WhatsApp chats, which I was going through, and some of them I able to reply, some of them are still pending, because there are questions like you have to cover discriminant analysis, somebody says you have to cover Poisson regression, also somebody says this and that. You see, it is not possible to cover all the topics, but whatever is written there, maybe except discriminant analysis, I'll try to cover all the topics. Don't worry at all. So there is a lot of messages about the logistic regression. Somebody says you have to cover, somebody says no, not to cover. But I think logistic regression is the most important part as far as medical field is concerned. And we are conducting this workshop for MGM Medical College. I think I will cover logistic regression today, definitely. Okay, let me share the screen and then start. Can you see my desktop?
Yes, sir.
Is it visible? Okay, yes. All right. So yesterday we were looking into one sample t-test, then independent t-test and pair t-test. Now, straight away, I'll come to what is called analysis of variance. And analysis of variance is a very important technique in medical field, in social sciences, biological sciences. So straight away, let me go to what is ANOVA without further wasting any time. So we have covered all these topics, if you remember. I'm trying to compile in one slide so that I can send only simple slide to you. I will also explain you what is the meaning of p-value today. And let me come to analysis of variance. Okay, so basically analysis of variance, if you look at one-way analysis of variance, it's an extension of t-statistics. In t-statistics, we can compare maximum of 2 groups. You cannot compare 3, 4, 5 groups together, right? For example, let's say there are 3 groups of patients. They are receiving different chemotherapy treatments, and our response is a recovery time. And I want to compare whether the recovery time of all the patients groups are the same or there is a difference. So obviously, you cannot apply t-test because t-test is going to compare only 2 groups. But if there are more than 2 groups, then you have to adopt for the another technique. And that's why we come to the technique which is called ANOVA. And ANOVA is basically called analysis of variance. So what happens in ANOVA? There are many types of ANOVA. There is one-way ANOVA, there is two-way ANOVA, there may be multi-way ANOVA, and there may be MANOVA also, multivariate analysis of variance. Then there is ANCOVA also, analysis of covariance. So there are many, but it is not possible to cover all of them. But anyhow, I will cover one-way and two-way ANOVA and also the ANCOVA right in the beginning. You see, what is ANOVA? It is a technique which partition the total variation. Whenever data is there, variation will be there. So it partition the total variation present among the set of observation into number of components. Now, how these components are there, just wait for a moment. I will tell you that analysis of variance is dividing the entire variation present in the data into certain components. You just remember this. And in one-way analysis of variance is used to determine when there are any significant difference between the means of 3 or more independent groups, because t-test can compare only up to 2 groups, and if there are more, then we have to apply what is called one-way ANOVA. So first I will discuss one-way ANOVA. Let's say there are k groups. Let's say there are k groups, and suppose the k groups, that the responses of all the k groups or the mean responses of all the k groups are same. Then I can frame the hypothesis that H0 mu1 is equal to mu2 is equal to mu3 is equal to mu k. So if I'm not including mu3 up to mu k, then it is mu1 is equal to mu2, which can be handled with the help of a t-statistics, right? Here mu stands for the group mean and k is the number of groups. So it depends. You are comparing 3 groups, you are comparing 4 groups, whatever the number is. That you can do with the help of one-way ANOVA. Let me straight away come to the model of one-way ANOVA. See, in one-way ANOVA, there is always some kind of response variable, and it is a continuous variable. Let's say, look at this. Suppose there are K treatments, N1 and 2. As I mentioned, let's suppose there are K different type of chemotherapies. Am I audible?
Yes, sir.
Okay. Suppose there are K different type of treatments, and if you want to compare them simultaneously, right? So model is like this, that Yij, which is a response variable, is equal to mu plus alpha I plus Eij. Those who are not from statistics or mathematics, don't look this model as a very serious model. It's nothing but a response variable, and response variable is affected by the particular factor, where this mu is the overall general effect and Eij is called error. Let me give you an example so that I can make all the terms clear. What do you mean by Yij? What do you mean by mu and all this? See, this, if you are comparing K different treatments, then your alpha I will be alpha 1, alpha 2 and alpha 3. And Yij, maybe you can look at the recovery time of the patients, right? Now, there are certain things which are common to all the patients. For example, mu is called the overall general effect. Certain things are there which, for example, if they are in the same ward, I cannot say that this person is taking more oxygen than the other patient. There are certain factors which are common to all, right? For example, in agriculture experiment, so rainfall is common to all the fields, or soil fertility is common to nearby plots. So there are certain factors which are constant, which you cannot change. So we put that under mu. But this is overall general effect, which is same for all, whether it is any patient is from group number 1, patient is from group number 2 or group number 3. And alpha I denote the effect of the ith treatment. So if there are K treatments, so it will be alpha 1, alpha 2, alpha K. And I want to test whether the effect of all the K treatments, is it same or there is a difference. How I can check? Looking at this response variable. Response variable is, let's say, recovery time. You give the treatment number alpha 1, let's say treatment number 1 to one group of patients. And then there is an independent group. You give the second treatment, you give the third treatment, you give the fourth treatment, and there are, let's say, 4 groups. So basically my hypothesis will be that alpha 1 is equal to alpha 2 is equal to alpha 3 is equal to alpha 4. This I want to test. And if you have only 2 groups, you can test with the help of T statistics. Because there are more than 2, so I have to go for one-way ANOVA. So in this model, Yij is nothing but response variable like recovery time. If you are dealing with agriculture experiment, then this could be yield of a crop, where this, you can say certain factors which are constant like rainfall, soil fertility, they are constant, they are put under mu. And this is the type of the seed used, the type of seed number 1. Let's say there are 3 different type of seed, let's say, and you give for a particular group of fields, you give one type of seed, for second type of seed and third type. So you can transform this experiment to anything where the factor is involved and a response is involved. And we have to minimize this error. And basically our aim is to test this hypothesis. Okay, now you look at this diagram, that will make you more clear. Suppose I want to compare K groups or K brands. Let's say brand number 1, brand 2, brand 3. Let me give you a very simple example. Whenever we go to a market to buy, let's say, tubes or light bulbs, generally we have some kind of brand in mind, that I want to buy maybe, let's say, Sylvania Lakshman or Philips or Bajaj, right? This is why, because we always think that if we take a brand of a standard brand, then their life length will be good, right? So let's say B1 is first brand. Suppose I call this as Philips or Bajaj, whatever you want to call. And I want to test whether the mean life length of Bajaj, Philips, or maybe Crompton or Sylvania Lakshman, whether it is same or there is a difference, right? Now suppose I frame the hypothesis that there is no difference between the life lengths. So your H0 will be that mu1 represents the mean life length of the first brand, mu2 represents the second brand, mu k represents the third brand. So your H0 will be that mu1 is equal to mu2 is equal to mu3 is equal to mu k. This is your H0. Alternative will be that at least 2 of them may differ. At least two of them may differ, right? Okay. Now, in order to carry out this experiment, whether the life lengths are same or not, I need to do, I need to look at the response. Let's say I take 10 tubes from Philips and put them on test. Let's say this is, you see, one is fixed right in the beginning, one is fixed. That means all these tubes are coming from brand number one. Okay. And how many tubes I have taken? 1, 2, right up to N1. So total number of observations which I have taken from brand number one is N1. This notation is very important for you. So I have taken N1 number of tubes from brand number one. Brand number one may be Philips. Then look at now 2 is fixed. That means these observations are coming from brand number 2. Again, how many observations? 1, 2, N2. N2 number of observation and so on. So it is K is fixed. That means it is coming from the Kth brand. K, K, K. And this is NK. Similarly, this could be treatments. This could be type of seed used or whatever you want to say. Now, what is Y11? Y11 is nothing but when it was put on test. So obviously it will burn for certain hours. Let's say first tube burns for 722 hours. So your Y11 will be 720 hours. Let's say this tube burns for 623 hours. So that will be your answer, right? So these are your responses, and the responses from brand number one, say Philips, brand number two, brand number six. Hopefully you understood the problem, right? And how many total observations we have? N1 from this, N2 from this, and Nk. So total number of observation will be N, and N will be nothing but N1 plus N2 plus Nk. So this is your total N. Now, please try to understand what we are trying to do. Do you think that even if the tubes are coming from the same brand, they will be having the same life length? Although they are coming from the same factory, maybe same material, maybe same person is operating the machine, but still there will be a variation. So there will be a total variation present in your data. Variation will be present in the data. So there are two types of variation. One is between the brands, between different brands, variation will be there. Another will be within the variation, within a particular brand, right? For example, this from the same brand, it burns for 730 hours. This is for 623 hours. So there is a variation. But which type of variation you likely to be more? Which are coming from different brands. So between variation will be more, and within variation is likely to be small, because within they are coming from the same brand. So it will be within variation you can expect to be smaller, and between variation to be larger. Actually, if they are coming from the same brand, same machine, same material, there should not be any variation, but variation is always there, little bit, right? So within variation is sometimes also called error. So what is ANOVA? Analysis of variance is a technique which divides the total variation, this total variation present among the set of observations, into two types of components here. The first component is between variation, that's a between the brands. Another is within the variation, within the particular brand. And if you add between plus within, you will get the total variation. So maybe if you have a copy and pen, you can write down TV, T for television, TV is equal to BW means between variation plus WV within variation. So total variation, which is TV, is equal to between variation plus within variation. So we can factorize, and this technique is basically called ANOVA. That it divides the total variation present among the set of observations into number of components, right? So we have two components for one-way ANOVA. And if we go to two-way ANOVA, there will be three components. Okay. So it will separate out and you will get this type of table actually. And I will show you how you will get this type of table. So let's say total number of observation is N. And if you are having, so your observation will be, degrees of freedom will be there. I will tell you the definition of the degrees of freedom for a moment. Please remember, if there are N number of observations, then N minus degrees of freedom will be there. If there are K number of brands, 1, 2, 3 up to K, then K minus 1 number of degrees of freedom will be for brands, right? This is what I have written here. You see, in this table, how many brands we are dealing with? K. So degrees of freedom, DF means degrees of freedom will be K minus 1. How many observations? Total observation N. So degrees of freedom is N minus 1. And between plus within will be total. So if I add this and this, you will get n minus 1 because k and k will get cancelled and you will get n minus 1. Or other way around is you will not have to compute the degrees of freedom for within because this can be obtained by subtraction. So n minus 1 minus k minus 1. So minus will become, so n minus 1 minus k plus 1. So 1 and 1 will get cancelled and you will see n minus k. So that's why degrees of freedom for this is k. So it will give you the sum of squares due to treatments. This SST is called sum of squares due to between samples, or you can say sum of squares between samples. This is sum of squares within samples, and this is the total sum of squares. And then we compute mean sum of squares in our table. What is a mean sum of square? You divide this factor by the corresponding degrees of freedom. You will get mean sum of squares due to treatment. MST is called mean sum of squares due to treatment. Similarly, if I divide sum of squares due to error by the degrees of freedom, I'll get mean square error. MSC is called mean square error. So you will get this table when you analyze the data. And if you take the ratio of the two, I mean MST divided by MSE, you will get F value. And corresponding to F value, you will get P value. You will get P value. And you have to just check whether your P value is less than 0.05 or more than 0.05. Let's say it is more than 0.05. Then you have to accept H naught. And what was our H naught? That mu1 is equal to mu2 is equal to mu k. That means your inference is that the life length of all the brands is same. Now, if the life length of all the brands is same, experiment is over. You go to market, buy any brand because the life length of all the brand is same. So experiment is over. All right. Hopefully you understood this part. But suppose I reject H naught. Suppose you reject H naught. When you will reject it, P value will be less than 0.05. If P value is less than 0.05, suppose it is coming out to be 0.0001. So I have to reject H naught. Now, if I reject H naught, the conclusion will be that there is a significant difference between the brands. Now, the next question comes, which brand is better? Either Philips is better, or Crompton is better, or Sylvania is better. And followed by which other brand? Whether it is followed by Bajaj or it is followed by some other brand. So I have to check that also. And to do this kind of analysis, we further apply post hoc test. And generally Chaffe's or Tukey's test, they will give you similar results. This I will discuss. So hopefully the crux of the one-way problem, one-way null variance is clear, that we will get F value. And if F value is non-significant, experiment will be over. But if F value is significant, then we have to further go for post hoc test. And it will give you a multiple comparison. It will compare one brand versus second, one brand versus third, one brand versus fourth. Then one is over, it will then take two versus one, two versus two versus three, and so on. So this is called one-way ANOVA, right? So let me cover the theory of two-way ANOVA and then I'll give you the demo. What is two-way ANOVA? See, as I mentioned, in one-way ANOVA, we are looking for in one direction only. If you look at the table, there is a variation between the brands basically. We are looking into only brands. There is one factor. But if you go for a two-way ANOVA, we have the problem like this. Let me go through the table. Let's say this is yield of a crop, and here you are using different type of fertilizers. A1 is one fertilizer, A2 is another fertilizer, and AP is Pth fertilizer. So P different type of fertilizers you are using. And these are, let's say, seeds. B1 is types of seed 1, B2 is type of seed 2, and Q different type of seeds. For example, this plot of land, this plot is receiving what? It is receiving fertilizer number 1 and seed number 1. But look at this one. Here fertilizer is changed to 2, but seed is again 1, right? Similarly, so all these observations. So this is yield of a crop when they are affected by two factors. Which factors? One of them is alpha I. Alpha I means effect of different type of fertilizers used. And beta J means effect of different type of seed used, right? So basically, that's why Yij response. What is the response? The response is yield of a crop. So yield of a crop depends upon two factors. One of type of seed used, type of fertilizer used. That's why this is called two-way ANOVA. And same way, in case of, let's say, medical experiments, if this Yij is a response of a treatment and you are giving 3 different type of treatments, let's say alpha 1 chemotherapy 1, 2, 3, and beta J looking into 3 different age groups. Let's say those patients who are into age group of 30 to 40, those who are in between 40 to 50, and third is who are more than 50, right? So beta J correspond to age groups, alpha J correspond to treatment, and this is your recovery time. So that sort of problem you can tackle with the help of what is called two-way ANOVA. So two-way ANOVA is basically extension of one-way ANOVA when your response variable is affected by more than one factor. That means it is by 2 factors. If it is affected by 3 factors, then we have to go for a 3-way ANOVA, 4-way ANOVA, and all this is called multi-way ANOVA. Now, here we have to test 2 types of null hypothesis. One is the effect of different type of, let's say, these are the different type of fertilizers, effect of fertilizers, alpha 1 is equal to alpha 2, that the effect of fertilizer is same or not. And second hypothesis will be that the effect of seeds is same or not. So basically we are testing 2 types of hypothesis. So same way we can extend one-way ANOVA to two-way ANOVA. Instead of, now total variation will be there. Now how many total observations are there? P observation here, Q observation here. So total cells will be P into Q. So P into Q. And degrees of freedom is always 1 less, PQ minus 1. And how many different types of fertilizers are there? P. So degree of freedom will be P minus 1. How many seeds are there? Q. So degrees of freedom will be Q minus 1. And this minus this plus this, if you add, you will get this P minus 1 into Q minus 1. So in two-way ANOVA, you will get basically one additional factor. The total sum of square, then sum of square due to levels of A. I call this as levels of A. Then sum of square due to levels of B, where levels of A stands for the different type of fertilizers. Level of B stands for different type of seeds. And again, this is error which can be subtracted. I mean, this minus this plus this, you will get this thing. And then again, you compute the mean dividing by the degrees of freedom. So this divided by this divided by this, and this divided by this degrees of freedom. Now you will get 2 hypotheses here. One will be for F1 for testing the effect of fertilizers. Another will be F2 for testing the effect of seeds, right? So one will be this divided by error. We always divide by error. Second will be this divided by error. Error will remain in the denominator, right? So this you can see mean sum of squares due to levels of A divided by mean sum of squares due to error. So that is the F1. Second will be mean sum of squares due to the type of seed or levels of B divided by the mean sum of squares. And then corresponding P value will be there. And just you have to check whether your P is less than 0.05 or P is more than 0.05. If P is less than 0.05, we will reject this. Again, if P is less than 0.05 for the second one also, we'll reject this. It may also happen that for the first hypothesis, the P may be greater than 0.05. That means there is no difference between the fertilizers. And in the second case, it may be less. In that case, you have to say that there is a difference between the seeds, but not in fertilizers. So post hoc will be carried out only for seeds then, not for. Post hoc has to be carried out whenever your result is coming out to be significant, right? So this is called two-way ANOVA. This is called two-way ANOVA. So I have explained you one-way and two-way ANOVA, and let me go back to the practical then of one-way ANOVA and two-way ANOVA. So I'll open this some data set and show you one-way ANOVA. Okay. So this is one-way ANOVA, which is an extension of T statistics. Okay. Now it's almost the same experiment. How you have to feed your observations. I think there is one of the assignment also, and somebody has asked me that there are values under A, values under B, values under C. I have given you one assignment. I think this is assignment number maybe 3 or 4. I don't remember. Maybe 3 or 4, whatever it is. Oh, no, I think it is assignment number 4, where there are certain values under A, certain values under B, and you have to apply one-way ANOVA. So you write down A values first of all, and in front of this, that they are coming from A, you write down 1, 1, 1. This is your assignment. Then After that, you write down B values below that, and you write down 2 that they are coming from B. And after that, you can write the third, C. So we have 4 brands here. This is how I have written that these are the life length from different brands. So if they are coming from one brand, so later on, I have gone to the variable view and change the labels that one represents, in your case, one will represent A, 2 will represent B, 3 will represent C. But in my case, one represents Bajaj, 2 represents Philips, 3 represents Crompton, and 4 represents Sylvania. Okay. So I have 4 different brands, and these are the life length. And what I want to test? I want to test that these are the labels I have given later on. So first 11 tubes, they are coming from Bajaj. Next few tubes, they are coming from Philips, then Crompton, then Sylvania. This is how you have to feed the data. There are only 2 columns. So this is a factor or a group, or this is your response variable, or a dependent variable, you can say, the left hand side of the one-way ANOVA model. Okay. Now I will carry out the analysis. So what we are testing? We are testing that the life length of Bajaj, Philips, Crompton, and Sylvania, they are same. That is our H0. Against the alternative that at least 2 of them will differ. And then we will see which ones differ. Okay. So here is analysis. Go to compare means, go to one-way ANOVA. And in one-way ANOVA, your dependent variable, which is on the left-hand side, is Yij. So that is the life length. Your brands is your factors, right? And then simultaneously, let me apply post hoc test also. Although post hoc test is required only when your result is coming out to be significant. But let me apply just to save time. And there are many post hoc tests. But the best test is one of the Sheff's test is good, and Tukey's test, because they are in, I will tell you why they are better than many others. So Sheff's, and if you are from the biochemistry department, most of the biochemists, they are using SNK test, which is called a student Newman-Kuhl's test. And sometimes in our category, there may be a control group, and other 3 may be experimental group. And if this is the case, you can apply this Dunnett test in that case, right? You can apply it, but in our case, there is no control category. Okay. And then press continue here, and then go to options, and in options, you click on to descriptives and means plot. That's it. So this is one-way ANOVA. It's very, very simple, and it will give you the result. So it will divide the total variation present among this into basically between brands, within brands, and the total. And you will get F value and P value. So here is the result. Press OK. So I'll show you the result. For example, first is just a descriptive table. How many tubes from each brand? Bajaj 11, Philips 9, Crompton 9. Total number is 37. So total number is 37. So you can see the degrees of freedom in ANOVA table is 1 less. So 36. And how many brands are there? 1, 2, 3, 4. And how many between groups? Between groups, the degrees of freedom will be 3, because 4 groups. So degrees of freedom will be 3. So this is between variation. This is within variation. This is the total variation. If you add the 2, if you add these 2, for example, 6 plus 6 is 12. Then 5 plus 3, 8 plus 1, 9. So you have this total. So this plus this is this, right? And this plus this is also this. And if I divide 3694 by 3, you will get this. If I divide 35 this by 33, you will get this. And if I divide 1231 by 107, you will get this. And based on this, there is a p value. So all these are related to each other. All these are related to each other. The higher the f value, smaller will be p. So here f value is quite large, 11 point something. So that's why p value is small. There is inverse relationship between p value and the value of the test statistics. Today I will tell you what is the meaning of p value. Okay. So what do you think? P value is less than 0.05. So I have to reject H0. I have to reject H0. And if we reject H0, that means I can give the inference that there is a significant difference between all the 4 brands. There is a significant difference between all the 4 brands. Okay. Now you look at which ones differ. Then there is a post hoc test. For example, if I'm using Tukey's test or Sheff's test, you will get almost the same result. For example, first of all, it will keep the first one. First one was Bajaj. Compare with Philips. You see, Bajaj, this is called multiple comparison. Since H0 is rejected, so Bajaj with Philips, this is a p value. It is more than 0.05. So that means non-significant. So there is hardly any difference between Bajaj and Philips. What about Bajaj and Crompton? This p value is again less than 0.05. So there is a significant difference between Bajaj and Crompton. What about Bajaj and Sylvania? Again, p value is more than 0.05. So again, there is a difference. So this is how you can check. So once Bajaj is over, it will keep the other one. And the p value of either you compare Bajaj with Philips or you compare Philips with Bajaj, p value will be same. So you just have to look at this column, right? But if you look at the Crompton, Crompton differs from Bajaj, significant. Crompton, if you compare with Philips, again, there is a significant. So Crompton seems to be different. So this is either you apply Tukey's test or you apply Sheffe test, you are getting the same result. So this is a multiple comparison. Rather than looking at this complicated table, there is a simplified table, and this is called homogeneous grouping. So it will give you homogeneous grouping actually. So it will put those groups into one subset which are very similar if there is no difference. For example, we have seen that in Sylvania, Bajaj and Philips, there is no difference. You can see from here, right? So they have put into one group. This is one group. While Crompton is different from all, it has put into another group. Now what is and the same result is true for Sheffe. Either you apply Sheffe or Tukey. So this homogeneous subsets is easy to look into rather than the other complicated table. So it basically says that the mean life, what is the mean life length of Crompton? It is only 59. And we want to buy that brand which is having the maximum life length so that it can burn for maximum number of hours, right? But there is hardly any difference between, look at the, which brand is better, you can see. If you look at Sylvania, it is 76. If you look at Bajaj, 82. If you look at Philips, 84.7. So that means which brand is having the highest life length? Because we are looking for highest life length, right? So it is 84. 84. That means Philips is giving better result. Followed by what? Followed by Bajaj. Followed by Bajaj. So there is hardly any difference between Bajaj and Philips. Let's say Philips, I want to buy 1000 tubes and I went to a market and there is a 20 rupees difference in Bajaj and Philips per tube. And I want to buy 1000 tubes for MGM Medical College, right? Now 1000 tubes, 20 rupees per tube. So I can save a lot of money because there is hardly any difference between. So I will go for Bajaj rather than going for Philips. Hopefully you understand. Let me just explain you what is the meaning. जब लेडीज सूट लेने जाती है ना मार्केट में, तो दो तरह के सूट दिखाई देते हैं. एक तो जो उनको पसंद आता है, दूसरा जो घर पे आता है. मेरे ख्याल से मेरी बात समझ आ गई. क्योंकि कई बार ऐसा होता है कि जो पसंद आता है वो बहुत महंगा होता है, उसको हम अफोर्ड नहीं कर सकते, फिर हम जो सेकंड चॉइस जो प्लान बी होता है उसको ले लेते हैं. तो यही चीज यहाँ पे हो रही है कि अगर हम फ्लिप्स को अफोर्ड नहीं कर सकते, देन वी कैन गो एटलीस्ट फॉर बजाज. If you cannot afford even Bajaj, you can go for Sylvania. But if you are opting for Crompton, then obviously you are on a loss. So comparison is between two because they are very close. All right. So answer to this is given by one-way ANOVA. So that is all about one-way ANOVA. It will give you the comparison. So I have given you about the one-way ANOVA. Let's come to the two-way ANOVA. Let me come to the two-way ANOVA. Open the another data set file for two-way ANOVA. Two-way ANOVA. Let me take up this example. Two-way ANOVA. So this is the data. You see, let me show you the data first of all, which kind of data we are dealing with so that you can understand what I'm doing actually. This one of the exercise I have already given you, and this type of data you have, or this type of table you have. I think in one of the assignment number 4, you have two-way analysis of table where there are certain figures here and certain figures here. But the data which I am solving is this is, let me tell you the experiment, what kind of experiment we are dealing with two-way. For example, when there is a voting machine, I mean, votes are polled after every 5 years, you know, then there is a voting machine and special training is given for voting machines. And people are hired from different departments of the government. And they are given 2-3 days or sometimes 4 days of training, how to operate this machine. And people learn the process. Now they have the three methods of teaching: method A, method B, method C. All right. And there are age groups, 5 age groups: less than 20 years, 20 to 29 years. So these are the 5 age groups, and these are the number of hours. So if you teach by method A and you are teaching to less than 20 years, then it took only 7 hours to make them understand what is the right. He takes 7 hours to learn. But if you teach by method B, again the same person, it takes 9 hours, right, in the same age group. Not the same person, some another person, but age group is same. Similarly, if you teach by method C, it takes 10 hours. So actually, this is a response variable, hours, number of hours, which is affected by two factors. Number one, method of teaching. This is a method of teaching. And it is also affected by the age group, in which age group you are. So basically, the response variable is affected by two factors. One is method of teaching, another is age groups. So it's a two-way problem. Now, what is our problem? We want to see whether all the methods are same or all the age groups are same as far as their learning is concerned, or there is a significant difference. So that's why this is called two-way ANOVA. And if you look at the SPSS file, you look at the first row, 7, 9, 10. This is how I feed the data. Please remember, this is 7, 9, 10. And 7 is coming under method A, 9 is coming under method B, and 10 is coming under method C. And you can see this word file. See, this is 7 is under method A, method B, method C. But they are all coming under what? They are all coming under less than 20, right? So they are all coming under this less than 20. Okay, second. So this is factor number 1. This is factor number 2. Similarly, the second row you can pick up 8, 9, 10. So again ABC, but they are coming under 20 to 29, 20, this group, age group. Similarly, so this is how you have to feed the data. Hopefully you understood this part. This is one of the exercise. So initially what I did actually, I just put for method ABC as 1, 2, 3, again 1, 2, 3, again 1, 2, 3. And for age groups, I have, because they are coming from the same age group, so 1, 1, 1. And then label. Later on, I have labeled this one as less than 20 and label this as method A, this as method B, this as method C, right? So this is what I did. So it's a two-way ANOVA. One is by method, another is by age group. So how to carry out the analysis? So go to analysis. Now, if you go to compare means, there is no two-way ANOVA. There is only one-way ANOVA. Then people think that there is no two-way ANOVA in SPSS. No, you have to go to general linear model. This is called general linear model, and this is called a univariate. Now, if you go to univariate, and in univariate, your dependent variable is this time. You take this time. What are the fixed factor? You have two fixed factors instead of one. In one-way ANOVA, you have only one fixed factor, but here you have two. One is method, another is age groups. So take both of them to this side, right? And then important part in two-way ANOVA is you must click on to model, please. Otherwise, you will not get the result. You can note down the steps. You click on to model, and in model, you click on to custom. There is a full factorial, there is a custom. You click on to custom, then they take this method on this side, also take this age group on this side, and change this type. We cannot study the interaction. Interaction has to be studied only if there are more number of people in age, in age method. Let's say I take 6, 7 people who are being taught by method A. I take 10 people who are being taught by method B. In that case, interaction is valid. If there is one observation, then interaction is not valid. So I have to click on to main effects. These steps are important. Otherwise, for the assignment, you will not get the result. Note down the steps, please, and press continue. Press continue. Then come to plots also. In plots, you can, let's say, method on age group on this side and go on adding. Then continue. Go to post hoc test. Let's say I don't know whether the method will be rejected or this will be rejected, but let me just apply just to save time. Method age groups. Let me apply this time only Sheffield test. You can apply Sheffield or 2K, you will get the similar results, right? Press continue. Then comes to the options, and in options you have to click on to descriptives. You have to click into, that's it, only descriptive statistics for method and for age groups. That's it. Nothing else. Press OK and you will get the result. So if you want me to repeat the steps, let me just repeat for two-way ANOVA. I have just clicked onto this, go to general linear model. Those, please note down the steps. Go to univariate, then take this variable on the dependent variable. You have two fixed factors. One is method, another is age groups. Click onto model. In model, click onto custom. Custom, you take this method on this side, age group on this side. Click onto main effects only, main effects. Continue from here. Then click onto plots, and in method here, age groups here. And you can draw the another plot also. Take age group on x-axis and method on y-axis, and you will get another plot. Continue, and then in options, you just need to look at the methods on this side, age groups on this side, not the overall, and go for descriptive. That's it. This is what you require. So I repeated all these steps so that you should not make mistake. Continue, press OK. And here is the result of two-way ANOVA. First of all, it will give you that how many observations under each method, how many people under each age group. We have already seen that there are 3, right? How this 5 and 3 is coming, you can see the word file. This 3 is coming because 1, 2, 3. And how this 5 is coming, 1, 2, 3, 4, 5. So this is column wise 5, row wise 3. That's why the table is there. Okay, let me close this. Now you understand this part. Okay, so this is not important. It is simply mean SD table. Why this SD is not computed? Because there is one observation only. So SD cannot be computed for one observation. This is one, but it can be computed for 5 number of observations. This is the most important table which you need to understand. Now, how many methods are there? 3 methods. So just at the degrees of freedom will be 2, 1 less. So this is just look at method and its significance. So this is the F value, this is the P value. So it says that P value is less than 0.05. That means there is a significant difference between the methods. One thing is clear. Let's look at age group. How many age groups were there? 5 age groups. So degrees of freedom will be 4. So this is 4. And it says again, it is coming out to be significant. P is less than 0.05. So that means there is a significant difference between methods, teaching methods, and also there is a significant difference between age groups. Now question is, I have to go for post hoc analysis. Otherwise experiment would have been over here. You just quote the result that there is no significant difference. All the methods are same, all the age groups are same. Okay, but I have to go further now. And further is a multiple comparison. And here is a multiple comparison. It's just coming up. I think I haven't clicked the multiple comparison option. Made a mistake. Okay, let me click on to multiple regression. Sorry, multiple options. General linear model, univariate, and there I have to go for post hoc. I didn't click actually. Post hoc for this, post hoc for this. Cheffe. In the first case I clicked. When I repeated actually I forgot. Anyhow, doesn't matter. Press continue, you will get the same result here. So again, so these are coming out to be significant. Now you go for multiple comparison. In multiple comparison, first of all, method A versus method B. Look at the P value. There is no difference. Look at method A and see there is a difference because P is less than 0.05. Similarly, you can make other comparison, but better way is to go for homogeneous grouping. Now, homogeneous grouping says that if you teach by method A, it takes 9 hours to learn. If you teach by method B, it takes 9.6, but there is hardly any difference between the two because it is non-significant. But if you teach by method C, it takes 11.6 hours. So obviously that method will be better, in which the minimum time requirement is there, not maximum, minimum time is there. So which method is better? Method A. So you can conclude that method A is better, followed by which method? Method B. But let's say method A requires very sophisticated techniques, like you need to go for a Zoom meeting, you have to go for the overhead projector, you have to go for LCD and computer and everything, right? While method B requires only blackboard and chalk, and you have seen that there is hardly any difference. So to cut off your cost, maybe you can adopt for method B rather than adopting for method A. So that sort of decision can be taken in two-way ANOVA. So but here method A is better. Let's say which age group learn fast, which is having less time. So less than 20, if you compare with 20 to 29, there is no difference. significance value is more than 0.05. Here also more than 0.05 if you compare the next age group. Here also. But if you compare less than 20 and more than 50 age group, there is a significant difference. So you can make all comparison. The better way is this table. Now you can see there is a overlapping also. If there is a overlapping, you just double click on this. You double click and remove cut from here. This will be out of the picture. So this is here. You retained in the first. That means these are the age groups. So which age groups learn fast, which takes less time? Let's see how many hours, more than 50 years. Those people who are more than 50 years, they take 12.3 hours. And which age group learn faster? Less than 20 years. So younger people, they learn fast. So less than 20, followed by which, let's say it's a very sophisticated machine and you don't want to give this to a child who is less than 20 years. Maybe you can go for this one that I want to hand over to people who are between the age group 20 to 29, because 8.6 and 9, there is hardly any difference. So that sort of decision can be taken. And this is a graphical representation. Let's say I teach by method A, method B, method C. So less than 20 is this blue line. So less than 20 learns fast. And this is yellow line is here. That means more than 50, they learn very, very slow. And similarly, you can look for this one method wise. Which method is better? Method A is better in each age group. And this is for method C is worse. Okay. So this is called two-way ANOVA. So I completed one-way ANOVA. I also completed two-way ANOVA. Now straight away, I am moving on to a very important part, which everybody has demanded. And I also want to cover, which is called logistic regression. Then I'll take up your question and before proceeding further. So logistic regression is a very, very important technique. It will take 10 to 15 minutes. Please bear with me. It's a very, very important technique, better than discriminant analysis. Discriminant analysis is applicable to only few situations, but logistic regression is applicable to many situations, particularly in medical field and social sciences also, where you are looking for some kind of risk assessment. So let me open up one file just to give you some idea about logistic regression. Let's just go through quickly this slide. So logistic regression, we will see what do you mean by odds ratio? What do you mean by risk ratio? What is the sensitivity? What is specificity? And I have to teach you ROC curve. So ROC curve will also be covered in this. So if you are with me for next 1.5 hours, maybe 1.5 hours, you can say, I will try to cover most of the topics. I think all the topics if you are with me. But if you are feeling tired, then sorry for that. Okay, let me start with the logistic. See, logistic regression comes from logistic function. Logistic function is from a mathematics. You see, this, if you plot this equation, Px is called probability in terms of x, right? It's in terms of x, where x is a risk factor and this is x1 is one risk factor, x2 is a risk. Just try to understand this. I'll not go into mathematics. You see, whenever we plot this function, it will be plotted like this. It is always, now its value y, because this is y actually, this y will take value between 0 and 1. So this, the minimum value of this graph will be, it will start from minus infinity, but look at the value. Value is between 0 to 1 on y-axis. X-axis may be minus infinity to infinity. Now, let's say y is my risk factors. Suppose when a child is born, initially he is having some kind of risk when he is newly born. But let's say after 5-6 hours, the risk go on reducing because immunity is developed, right? So epidemiologists and statistics people, they thought of that y can be considered as a combination of risk factors. Suppose there is no risk, the person doesn't have any problem. If person doesn't have any problem, what is the probability that the person will die? It's almost 0. So this is when there is no risk. That means we start from minus infinity. Probability that a person will die will be almost 0. Almost 0. But let's say when the age grows, let's say person has developed diabetes. So one risk has developed. Person has developed blood pressure. Second risk has developed, that is x2. Person has developed kidney problem, x3. Person has developed liver problem, x4. So there are k risk factors now. So as the risk goes on increasing, obviously the probability that a person will likely to die will go on increasing. And a time will come when the person is ultimately die. 100% risk will be there if you go on adding the risk factors. And this entire picture is captured in this graphics, and that's why this is called a logistic regression. So logistic regression comes from a mathematical equation. Mathematics people, they have left this here only, but statistics people, they have picked it up, that it can be further classified as a combination of risk factors. When there is no risk factors, the chance of dying will be very, very low. But as you increase the number of risk factors, the chances that a person will die will be more and more. Hopefully you understand the problem of logistic regression. Now, if you look at this equation, y is equal to beta naught, beta 1, x1, it is same thing as multiple regression. It is same thing as multiple regression, right? But here there is a big difference. In logistic regression, in multiple regression, y, x1, x2, xk, they were all measurements. You remember IQ was measurement, then mental age, depression score, convolution, all were measurements. So but in this case, dependent variable in logistic regression is always dichotomous. It's a binary variable, either 1 or 0. That means person is having coronary heart disease or they are not having coronary heart disease. Or you can say person is dead or it's alive, or a student is present or absent, or the bank is giving the credit card, there is a risk or there is no risk, right? So such type of thing is covered under this. So you can apply in different situations. Hopefully understood the problem. And moreover, the independent variable, somebody was asking, you cover dummy variable also, you cover this and that. Dummy variable, I am not covering here, but here category, x1 could be categorical also, x2 could be categorical, x3 could be a category, or it could be continuous also. It all depends. So that is a flexibility logistic. Dependent variable is always dichotomous, but independent variable may be categorical variable or continuous variable. So that's why the logistic regression is important, and I want to cover this. So I will not go into details of this. That is the equation I have explained. I'll take up simple example and explain you the crux. Let's say I look at the application part. Suppose there are 3 risk factors. One of them is age, another is gender or sex, another is cholesterol level. And we want to predict the risk of developing a coronary heart disease. So whether the person is having coronary heart disease or not having coronary heart disease. So let's say Z is called coronary heart disease. And your first risk factor is X1, that is age. Age is a continuous variable. It is not categorical. So age is exact years, like 55 years, 60 years, 70 years. Then gender. Let's say gender is a categorical variable. 0 is male and 1 is female. Then cholesterol. Cholesterol can again be measured. Cholesterol can again be measured. I mean, cholesterol level, you know, it can be measured. And it is a continuous variable. So one of them is categorical, 2 of them are this. And then I can develop this equation, y hat is equal to this thing. And these coefficients can be estimated from the given data, right? Let's say what kind of data I will be having. I will be having value of Z, whether initially I will come to know whether person is having coronary heart disease or not having. If it is having, I will put this as 1. If not having, I'll put this as 0. Then age, first column, I mean second column, X2 the third column, and X3 will be the fourth column. This is how the data will be, right? So risk factors, you can have more risk factors like smoking you can include, diet you can include, exercise, alcohol use, all these, I mean, you can add X4, X5, X6. So this equation can go on depending upon your problem. All right. Okay. Now suppose I get this equation. So I analyze the data of 609 people and I get this equation. So this is X1, this is X2. X1 stands for age. Let's say I want to compute the predicted risk of a person. So without knowing what is happening, let's say, because this, if I put in this equation, which is a probability of Z, this value will always lie between 0 and 1. You see, this value will always lie between 0 and 1. Always between 0 and 1. Right? So let's say I look at a person who is 65 years of age. So X1 will be 65. This is from the given data I obtained this. So let's say I want to compute a risk of a person. What is the chances that a person is having coronary heart disease? Suppose his age is 65. He is a male. Male is, I think, let me see what is the code for male. Male is 0. So I'll put this as 0. And then X3. X3 is cholesterol. So I note down the cholesterol level, put it here. And suppose I'll get the value of Z. And you substitute the value of Z or Y. This is basically Y in this equation. I'll get the risk of the coronary heart disease. And this will be between 0 and 1. Suppose this value is coming out to be 0.73. That means the person whose age is 65 and a female and cholesterol level is this much is a 73% chance of developing a coronary heart disease, right? So this is called logistic regression. So here is odds ratio. So odds ratio is very important in logistic regression. Odds ratio, first of all, for example, let's say, suppose that the probability of success is 0.8. Then the probability of failure will be 0.2. So if you look at the odds of success, it will be 0.8 divided by 0.2. So it is 4. Look at the odds of failure. Odds of failure will be opposite. 0.2 divided by 0.8. So it is this. And what is odds ratio? You divide this by this, you will get odds ratio. So you will get odds ratio. Let me give you a simple example of odds ratio. Suppose that 7 out of 10 females are admitted to a medical school, while 3 out of 10 males are admitted, right? 7 out of 10 females are admitted and 3 out of females are more intelligent, right? So look at the odds of female. Odds of female will be 0.7 divided by 0.3, this much. Odds of male will be 0.3 divided by 0.7, this is this much. And if you take the ratio of these two odds, that will give you odds ratio. So if I take this divided by this, odds ratios, what is the meaning? Meaning is, thus the chances of that the females will be admitted to a medical school are 5.54 times higher unmute. Okay. Can you listen to me now? Hello. Can you listen to me now?
Yes, yes, sir.
Okay.
Yes.
Look at the odds ratio. So odds ratio says that what are the odds that the female versus male. So what is the odds that it is 5.44 times higher that they will be admitted to the medical school as compared to males. This is what do we mean by odds ratio, and it is given in logistic regression. Then the another part, like in case control studies, because we are dealing with the medical people here. So in case control studies, let's say these are the cases and these are controls. So exposed, a number of cases are exposed when it is under the experimental group, under the control group. This is non-exposed. And then you look at the odds of each. Odds of this will be exposed will be, let's say, A by C. And in control group, it will be B by D. And when you take the ratio of these two, it will give you the odds ratio. So odds ratio is just multiplying A into D divided by B into C if you are having a 2 by 2 table. So in case control study, we generally compute odds ratio. It's very simple. In cohort studies, we compute risk ratio. We cannot compute odds ratio. I will tell you the reason, right? So in cohort studies, what we generally do, we have disease and not diseased. Here it is exposed and not exposed. A number of cases, B. So incidence of disease will be A over A plus B. Here it is C over C plus D. And if you take the ratio of this divided by this, you will get risk ratio. Oh, sorry, this is called relative risk. So in cohort studies, we compute relative risk. And in case control studies, we compute odds ratio. Okay. So now if odds ratio is 1, that means there is no association. That means the males and females, they are having equal chance. You can say like that. But if odds ratio is more than 1, then females are having more as compared to males. And if it is less, then it is other way around. So that is the interpretation of odds. And based on this odds, then there is a very important curve, which is called ROC curve. And ROC curve is called receiver operating characteristic curve, which is used in medical sciences. So I will cover this topic because it is listed in our topic. You see, let's look at this quantity. Test has been conducted. So predicted positive by the test, predicted negative by the test, and it is actually, the person is actually having the disease or actually not having the disease. Okay. So just think like this. For example, the person is having the disease or actually is positive, and the test also says that it is positive. Predicted test also says that it is positive. So this is put under, that is called true positive. It will be a number. It will be a number. So this is called true positive. Because why true positive? Because test says it is positive and actually the person is also positive. So it's a true positive. Let's look at true negative. The test says the person is actually negative, let's say COVID negative, and the test also predict negative. Then it is a true negative. Then it's a true negative, right? So this is true positive and true negative. Remember like this. Now, let's say the person is actually negative. The person doesn't have a COVID-19, but the test says that the person is having the disease. So it is a false case. So this is called false positive. This is called false positive. Similarly, this is called false negative because the person, let's say person is having the COVID-19, but when the test was conducted, it says that person doesn't have. So this is called false negative. So this is diagonals are true and off diagonals are false. You can remember like this. And if I take this plus this, then this is called predictive positive. This is called predictive positive. This is predictive negative. This is predictive negative. And this is actual positive. This is actual positive. This is actual negative. And if you take the this or take the sum of this is total n, right? And based on this, there is a ROC curve. So for ROC curve, we plot sensitivity and specificity. Now, what is sensitivity? Sensitivity is nothing but the true positive divided by the actual positive. That is called sensitivity. And what is specificity? Specificity is nothing but true negative divided by the actual negative. That is called specificity. Then there will be another value which is called probability predictive value, right? That will be TP over PP. And then probability negative predicted value, that will be this divided by this. So all these are given here. So sensitivity is this, specificity is this, and positive predicted value is this, negative predicted value is this. They are coming out of this table. Now you understood all the definitions. So you take the ratio, you will get sensitivity and specificity. Then once you have the sensitivity and specificity for each cutoff point, then you plot on x-axis the parameter which is called 1 minus specificity on x-axis, and on y-axis we plot sensitivity, and you will get ROC curve. And then we have to look for the area under the ROC curve, right? So here is the example of ROC curve. Let's say a receiver operating characteristic curve is an excellent way to compare diagnostic test. So ROC curve is basically for the diagnostic accuracy of, it will give you the diagnostic accuracy of your test, how your test is accurate. Obviously, if false positive and false negative will be small, test will be better. But if they are more in number, the test is not good. The test is not good, right? Let's say I consider two types of groups. Consider the data for serum ferritin as a test for iron deficiency anemia. So iron deficiency, anemia is there. These are the numbers with who are having iron deficiency, and this is without iron deficiency. And I take the serum ferritin level, say less than 15. So this is the number. 15 to 34, this is the number. So these are levels actually of serum ferritin. I divided into few. Let's say I want to compute sensitivity and specificity. And as you know, sensitivity and specificity to compute, I need only 2 by 2 table. Then and only then I can compute. So let's say I can convert this into 2 by 2 table. Suppose I take 34 as a cutoff. Now, if I take 34 as a cutoff, then I can club these two values, say less than or equal to 34 and more than 34. And I can club these values, these three values. Similarly, on this side, I can club these two values and I can club these three values, and with 34 cutoff. Similarly, I can take 64 cutoff, I can take 15 cutoff, I can take 94 cutoff, and this is what you will get. So if you take 34 cutoff, you add the first two and the last three. You add the first two and the last three. And based on this, you can compute sensitivity. So sensitivity will be this divided by the total. This divided by this plus this is 80 and multiply by 100. So you will get 80.2%. Specificity, this divided by this and this. So you will get 94 point this. This is for cutoff 34. Similarly, if I take 15 as a cutoff, so I'll get the sensitivity and specificity. If you take 34 cutoff, for 34 we have already computed, right? For 15 you can compute now. Take the first row as such and club the next, last, all the next of the rows. And similarly, I can, if you take 34, then you can take the sensitivity and specificity. You can compute, I mean, for 94 also. So these are computed values. Then you take 100 minus this, 100 minus specificity, 100 minus this will be this, 100 minus this will be this. And then you plot this figure on x-axis and this figure on y-axis, and you will get this plot. And this is called ROC plot, right? And it will give you the diagnostic accuracy of your test. If it's area under this curve, I will show you how to compute the area under the curve. If area under the curve is 1, which is hardly achieved, then you have a perfect test. There is no false positive, there is no false negative, all are on the diagonal. But if it is between 0.9 to 0.99, excellent test. If it is between 0.8 to 0.89, good test. If it is between 0.7 to 0.79, fair test. And if it is between 0.5 to 0.51 to 0.69, poor test. And if it is area under the curve is below 0.5, it's a useless test. You should not apply this test at all. So that is the idea of plotting ROC curve. I hope I have made all the things clear. Now I'll go for a practical demo. It won't take more than 5 minutes. So by 5:15, it will be over. By 5:20, sorry. So let me take up the ROC curve and as well as the logistic regression. So open up one file. Open up data file for logistic. Okay. So this is a real life data in one of my research papers. If you see American Journal of Ophthalmology, we have published this paper based on this data. And that is having a very high impact factor. The data is like this. The age of the patient, then there is a gender, and glaucoma, you know, glaucoma is a problem of eye. The central vision gets distorted, you know, and the person, he was operated by the doctor, and there are, I think 1, 2, 3 different type of surgeries. One surgery is called A, another is B, then there is another is C. So person has been operated by 3 different type of surgeries, A, B, and C. And then after surgery, the glaucoma again developed sometimes. And this is called post-PK glaucoma. Post-PK means plateaucrestoplasty glaucoma, right? And this is called post-PK glaucoma, whether the person has again developed the glaucoma or not. So this is your dependent variable. Either they have developed or they have not developed. So if they have developed, then it is 1. If they have not developed, then it is 0. Right? So you have to code like this only. If it is a higher probability that the person has developed, take this as 1, not developed as 0. And type of surgery, ABC. And history of glaucoma. If person is having history of glaucoma, that's a person is having history of glaucoma as 1. If they don't have history of glaucoma, I think it is 0. Right? History means the parents are having glaucoma, or maybe some ancestors having glaucoma, and it is a gender. This is a gender, male and female. So this is your, please try to understand, this is your dependent variable, which is dichotomous in nature. And all these are independent variable. This is continuous, this is categorical, this is categorical, this is categorical. And I will apply logistic regression on this and also plot the ROC curve. Also plot the ROC curve and show you the interpretation. Okay, so how this can be done? So please remember, this is your dependent variable and these are independent. This is X1, X2, X3, X4. Go to analysis, go to regression. There is no logistic regression here, but logistic regression is also called binary logistic. This is binary logistic. So click on to binary logistic, and in binary, what is your dependent variable? Your dependent variable is post-PK glaucoma. So take this variable here, post-PK glaucoma, and these are all independent variables. So press Ctrl A and substitute here. Now, do you think all of them will be important to predict post-PK glaucoma? Maybe gender may not be, or history of glaucoma may not be, or type of surgery may not be. So if you apply forward likelihood method is the best method in logistic regression. Forward likelihood method is the best method in logistic regression. And only those variables out of these four will be retained which are making significant contribution to predict post-peak glaucoma. So I'll apply forward LR, then go to save, click on to probabilities, group membership. Group membership will give you the authenticity of your model. How many cases have been correctly classified, right? I mean true positive cases and true negative. So it will give you. And from that, the ROC curve can be drawn. Continue. And then in options, you need to click classification plots, Hosmer-Lessman goodness of fit test. And this is called odds ratio. Wherever it is written confidence interval for exponential B in logistic regression, this is called odds ratio, right? And then continue. I think nothing else. Let me see. Yes, nothing else. Okay, just click on to OK and I'll explain you the output. So we have 212 I's and it says that your original variable no cases are this, yes cases are coded as this. And don't look for this table. This is not important. Then the most important table in logistic regression is, first of all, see this ominous test for model coefficients. Generally, they will come out to be significant. And step 1, step 2, step 3. If you have only 2 steps, that means out of the independent variable, only 2 variables are coming out to be significant. But if you are going up to 3 steps, that means three variables are coming out to be. So it will depend on how many steps have been taken. So this is one variable included on step number 1, second on step 2, and third. And after that, it has stopped because other was non-significant. Then 2 important things to look for logistic regression. One is Cox and Snell R-square. This is equivalent to R-square of multiple regression. This is equivalent to R-square of multiple regression. Then there is Negerkete R-square. This is equivalent of adjusted R-square in multiple regression. Now their values, you cannot expect these values to be close to 1. Generally, if it is close to 1, it's a very good model. But even if it is close to 0.5, because there are categorical variables, then even the model is good. So this value is close to 0.4. So seems to be good. Okay. Then the most important table is this. What is the authenticity of your model? Go to, don't look for step 1, just like we have seen in multiple logistic regression also. Don't go for step 3, go for step number 3. Step number 3 says that your authenticity of the model is almost 85%. This is an overall percentage. That means if I take, you see, in model, you already know people are having glaucoma or they are not having post-PK glaucoma values, right? But our model, when you put up all the values of 212 patients into your model, the equation I have shown you, then it says that model prediction is almost 85%. That means if you take 100 patients, almost 85% will be correctly classified by your model. And anything above 70% is always good in logistic regression. This is what it means. So this table is important. And this is a table. This is true positive. This is true negative. This is false positive, false negative. So these are the values. And based on this, I will plot now sensitivity and I will compute sensitivity and specificity and show you how to plot this ROC curve. And your final equation will be this, which can be used for prediction. So your Y will be equal to 4.980 plus 0.024 into age minus 2.067 into sex plus minus 5.428 into history of glaucoma, because these are significant values. Now there was one variable which was surgery, type of surgery. It has been excluded. Why it has been excluded? Because it says that its value is variables not in the equation. So type of surgery is not in the equation because its value is non-significant. So it has been automatically removed from the model. So model is based on only 3 predictors now. And model authenticity is almost 85%, right? So I'll go for the ROC curve. I have already saved the probabilities here. So these are the probabilities, right? And you can plot ROC. If you see under graph, there is no ROC curve here. Where is ROC in SPSS? Go to analysis and here is ROC curve. Go to ROC curve. Your test variable is the probability. This is your test variable now. So this is a predictive probability, PRE1, which I have saved. So take this on this side. State variable. State variable means whether people are having post-PK glaucoma or not. So that was your post-PK glaucoma variable. It says what is the value of a state variable. State variable means on which the event has occurred. So event has occurred when the people have 1. Then the glaucoma is there. So I have to write down this 1. Right? Then you click on to with diagonal reference, standard error and coordinates. That's it. In options, I don't think we require to do anything. No, nothing. Press OK and you will get a very nice ROC curve. So this is the ROC curve. You see, we have plotted the ROC curve from there, and the area under the ROC curve you have to see. This is the area under the ROC curve. You remember, if area under the ROC curve is between 0.9 to 0.99, it is excellent. And if it is between 0.8 to 0.89, then it is good test. So ours is a good test because model prediction is also almost 85%. If this would have been more than 95%, it would have been excellent. But anyhow, still it is a very good model, and this is how we can plot the ROC curve. And these are the, at each point, the sensitivity and specificity values which are computed at each point, right? So these are the sensitivity and specificity values. So with this, I have completed logistic regression, one-way ANOVA, two-way ANOVA. Now, before I proceed further, you can ask me questions up to this, please.
Yes,
now
sir.
I will take up factor analysis and cluster analysis. Yes, please.
Sir, first question is, what is the difference between fixed factor and random factors?
Very important question. See, for example, I have taken male and female as fixed factors, or type of surgery also as fixed factor A, B, C, because I know that there are only 3 surgeries. But sometimes what happens, let's say, how many varieties of mangoes are available in India? How many? Hundreds of. ये तोतापुरी आम है, ये दशहरी आम है, ये लंगड़ा आम है. I think everybody knows all these terms, right? So hundreds of varieties of seeds. So hundreds of varieties of seeds. अब आप 100 के 100 को तो टेस्ट नहीं करेंगे. Doing ANOVA table, you will pick up only few which are important. People are consuming. Generally दशहरी आम हो गया या लंगड़ा आम हो गया या इस टाइप के आपने 4-5 वैरायटी ली और उनको ही टेस्ट करते हैं आप. So randomly you pick up. Out of the 100 varieties, you randomly pick up only few. So if you randomly pick up only few, then they are not fixed actually. Somebody can take another 4, somebody can take another 4. So they are taken at random. So that for that purpose, we use random. If we have fixed, then we use fixed. If we have only 3, then we use fixed. If we have more than that, we want to select out of that, then we use random. Yes, please. Next question.
So next question is, while coding variables in logistic regression, how to decide reference variable coded as 0 and 1 or 1
Very
and
good. If the event is occurring, then it is always taken as 1. If doesn't occur, then it is taken as 0. For example, you are looking for coronary heart disease. Person is having, code this as 1. Person doesn't have, code this as 0. And similarly, you are looking for, let's say, in a bank setup problem. People are looking for whether, I mean, if I give credit card to this person, what is the risk involved? So based on certain factors like X1, X2, X3, the person is, what is the risk that the person will return the money or will not return? That means he is a defaulter or not a defaulter, right? So defaulter you put as 1 and non-defaulter you put as 0. But in case of gender, you can take even 1 and 2, you can take 0, 1. That doesn't make, because it is on a nominal scale. It is on a nominal scale. Yes, please. Next question.
next question is, sir, how to select reference category in logistic?
Already told. That 1
next,
is always taken if the event occurs.
yes, sir.
And 0 not.
How to calculate the predictive probability?
You will, if you try to complete by using your own by hand, you won't be able to do it because this is based very complicated calculations. So SPSS will do it for you. Why do you want to compute? It is based on a method which is called forward likelihood, maximum likelihood method. So it is computed according to that mathematical equation. You will not have to compute that.
Okay. So next question is, do we always use predictive probability for making ROC curve?
Yes, in this case, predicted probability will be there.
Next
In
is,
ROC curve, if you are plotting, yes, please.
sir, please explain predictive probability used in ROC curve. Same.
You see, predictive probability is
Uh,
nothing but, you see, what was our equation? Our equation was Px is equal to 1 over 1 plus e raised to power minus y. Now, y has been estimated from the data. And if you put the value of y, you will get Px. Px will lie between 0 and 1. And I have shown that for first case, I think probability was coming out to be 0.16 something. So this is how you put all the values, estimate y, and that put into that, and you will get the actual value of the probability, right? So this is how it is being computed. There is a mathematical formula to compute that. I have already shown the mathematical formula.
okay.
Yes, please.
Sir, next there is request, please explain interaction effect in two-way ANOVA with example.
I cannot do that because that will take another half an hour. I don't have time now. That will be in another workshop.
Next, sir, can we use logistic regression for questionnaires used to screen diseases such as obstructive sleep apnea?
Can you just repeat?
Can we use logistic regression for questionnaires used to screen diseases such as obstructive sleep apnea?
So if your dependent variable is 1 or 0 form, people are having disordered sleep or they are having good sleep, then you can. If you can categorize your dependent variable or a response variable in terms of two, either yes or no, I mean they are having or they are not having, then you can use logistic regression. There is no harm.
Okay, sir. So next question is, for the ROC curve analysis, predicted probability from regression equation is directly used?
Directly computed. I mean, you have to save that. I have saved that. There is an option to save. You go to save and it says that probability predicted. You click onto this. So it will be saved and then you draw the ROC curve for that.
Okay.
Yes, please.
So
Any
there
other
is one
question?
request to send the PPT of logistic regression.
That I will just try to include and then send it to you on WhatsApp group. Don't worry. Any other question?
I think all questions are clear, sir.
Okay. So there may be few other questions, but anyhow, because time is less, we are left with up to 6 and we can go up to 6:20 maybe. So I will try to cover now two important techniques. One of them is called factor analysis. Another is called cluster analysis. And then we will see how much time is left. And if you allow me to cover some other topics, then I'll cover. Otherwise, I will stop because everybody feels tired after learning so much. But anyhow, I never tires. I can take whole day, right? So you can ask me any questions even later on. All right. But I have given you enough literature to go for ANCOVA and MANOVA. I don't think I will be able to cover because it's a heavy dose. But let me cover two important techniques, and one of them is factor analysis and cluster analysis. Okay. Open up the same slide again. So this I will give you where factor correlation regression, logistic regression, estimation, testing, everything is there, right? And now we come to a technique which is called factor analysis. Okay, can we see the screen now of factor analysis?
Yes, sir.
Yes. Okay. See, what is a factor analysis? Factor analysis means you can reduce the dimensionality. It is called a dimension reduction technique. Factor analysis is called a dimension reduction technique. You can reduce the dimensionality of your variables. What do you mean by dimension reduction? So factor analysis is a general name denoting a class of procedures primarily used for data reduction or aggregation. I think some echo is going on. Hello. Is this okay now? Am I audible?
Yes, sir, no problem.
Okay. See, all the techniques. all the techniques which we have learned so far. I think if you mute all of them, some problem. Some connectivity issue. Is it okay now?
Sir.
Is this okay?
Yes, sir.
Okay. Now, in multiple regression, simple regression, logistic regression, we always have dependent and independent technology. I mean, there is a dependent and independent variables. There is no such thing like dependent or independent variables in factor analysis, right? So factor analysis is an interdependence technique. It depends upon one variable on another. Generally, factor analysis technique is used when the variables are correlated among themselves. Let me just repeat. Factor analysis technique is applicable whenever the variables are correlated among themselves. So let's say you look at the correlation matrix and variables are having high correlation. That means that is an indication that you can apply factor analysis. I'll take up one example to clarify, and then I'll straight away go to the demo part, and you can read this PPT from here. There are certain terminology. I'll try to explain everything. But with the help of this example, let me make it more clear. Now, you see, in any of the city, there are big malls these days. For example, in Chandigarh, there is a Elante mall. In Calcutta, I have seen hundreds of malls. I mean, there are malls everywhere. And people go to malls. And the owner of the malls is thinking that how I can improve upon the facilities of my mall. Can somebody guide me? Then he hire a team of educated people, let's say five, 6 people, and give them this task that you identify certain important parameters so that I can improve upon the facilities available in my mall, right? Then this team of researchers design a questionnaire, and there are certain, they take certain parameters. For example, cleanliness related to, they design a questionnaire, and some questions are related to cleanliness. How many questions? 15 questions are related to cleanliness. Cleanliness of lift, cleanliness of floors, cleanliness of toilets, and so on, right? So 15 variables are related to cleanliness. Then they look also for entertainment. If people go to mall, they also watch movie, and they also look for English movie, Hindi movie, or maybe Bangla movie, or maybe Gujarati movie, whether they are playing different type of movies or not. So there are 20 variables related to entertainment. Then there are 15 variables related to 36 to 50. They are related to parking. Parking means whether there is enough parking space or not, whether what is a parking charge is reasonable or not, whether they are, I mean, increased every hour or not, something like this. So there are 15 variables of this type. Then games for children. Generally, when you go to a big mall, there are games for children also. And 25 variables they are related to, whether they are having car also, whether they are having the other digital games also, and so on. So 25 variables they include, which are related to children's games. Then there are different brands. How many brands are available, whether they have Peter also, Peter England or something like that, different brands, Levi's and so on. So they look at 25 different kind of brands, whether they are available or not. And also they look for whether there is a McDonald's or Donut or maybe Sagar Ratna, or all these are there or not. So 20 variables they are related to food court. And in all, they design a questionnaire with how many questions? 120. So you have a questionnaire with 120 questions, right? Now the question important, which is the most important, is, do you think all the 120 will be important? Question mark. Do you think? May or may not be. Maybe out of 120, only 30 or 35 are important, or maybe only 20 are important. To look at the importance, which ones are important, which ones are not important, we apply a technique which is called factor analysis. Hopefully you understood this part. We apply a technique which is called factor analysis. Let's say I design a questionnaire, then hand it over to the respondent, and he or she has to fill it up and give you back, and you have 120 questions answer. You feed this into computer, and let's say there will be a variability. Let's say entire variability of your data is explained by only let 25 or 26 variables. Then why to unnecessarily include rest of the 95 variables into your model. You can exclude them and ask the owner that you must only focus on these 25, 26 variables. Rest of them are not important. He will also be happy. You will also be happy, right? So this is called data reduction. So factor analysis is a data reduction technique. Okay, how we carry out this? So we look into see, there is a variability present in your data. Don't look for the model. There is a variability present in your model. So there is a technique which is called principal component analysis. So it works on principal component analysis. So it's look into the linear combination of the variables. Let me just explain. Factor analysis, first of all, look into the linear combination of all the 120 variables, it will find out that linear combination which explains the maximum variation present in the data. Let me just repeat. First of all, what is a linear combination? Let's say there are two variables, x1 and x2. Then its linear combination is denoted by y, and y will be a1 x1 plus a2 x2, where a1 and a2 you have to compute from the data. Let's say it is 3 and 5. So your linear combination of two variables will be 3 into x1 plus 5 into x2. That's called a linear combination. But generally, we will not get 3 and 5. Their values between 0 and 1. I mean, one weight will be, for x will be, the coefficient will be between 0 and 1, y will also be between 0 and 1. Then I can see if it is closer to 1, this is having highest weight. For example, let's say x2 is having 0.9 and x1 is having only 0.2. Then x2 is more important than x1. Okay. So this is called weight. So it looks into the linear combination of 120 variables. Let's say out of these 120, let's say it identify 8 variables which explains 40% variation present in the data. And some variables coming out from cleanliness like V12, V8, V9, 3 variables from here, and let's say 5 variables from here. And they are explaining 40% of the variation present in the data. That is called first principal component, and that is called factor 1. That is called first principal component, and we call this as factor 1. Okay, which explains 40% of the variation. Then now we have already identified 8 variables which explain 40% of the variation. Now out of the 120, 8 will be removed. Now how many are left? 112. It looks into another, the linear combination of 112, and find out another combination which explains the next variation present in the data. Let's say the next variation explains 30% of the data, and it may be related to some parking space. It may be related to games for children. Some variables are picked up from somewhere, right? And let's say there are 12 variables which have been picked up, and it explains 30%. So first we have 8. Next, second factor we have 12. So 20 variable is explaining 70% of the variation present in your data, right? So now out of 120, 20 will be removed. Now you are left with 100. Again, it looks into the linear combination of 100 variables, right? And find out another linear combination. Let's say only 6 variables are there, and which explains say 20% of the variation present in the data. So first we have 8, then we have 12, and then we have 6. So in all together we have 26 variables, and these 26 variables, they are explaining 90% of the variation present in your data. That means the rest of the 10% variation is being explained by 120 minus 26. That means 94% of the variables they are explaining just how much? Only 10%. Why to include them into the model? They are not important. They are not important. So only 3 factors can explain 90% of the data. So this is called dimension reduction. So you have, I mean, reduced your 120 variables to how many variables which were important? Only 26. So you can focus on 26 rather than 120. So that's why this is called dimension reduction technique. And further, these 26 variables can be in terms of 3 factors only. Factor 1, which is called F1, factor 2, which is called factor F2, involving 12 variables, F1 involves 8 variables, and F3 involves 6 variables. So basically your score will be in terms of only 3 factors. And these 3 factors are generally uncorrelated with each other. The first factor, the second factor is derived in such a way so that it is uncorrelated with the first one. Third one in such a way so that it is uncoded with the first as well as second. And how these factors are coming? They are coming out of a matrix, and matrix calculate eigenvalues, and only those factors are important whose eigenvalue is more than 1. Please remember this. Only those factors are important whose eigenvalue is more than 1. Let's say factor 1 is having eigenvalue 4.3. It is important. Factor 2 is having eigenvalue. It will be displayed in the output. Eigenvalue 2.3. So it is important. Let's say F3 is having 1 point something. So it is important. But F4, the factor is having only 0.73. It is less than 1. So it will not be important. I will not consider this factor. This is how the factor selection goes. So if the eigenvalue is more than 1, you consider that factor. If it is less than 1, you should not consider this factor. That is one thing. Second, there are two important tests. There are two important tests in factor analysis. One of the tests is called Bartlett test of specificity. And Bartlett test will give you the value has to be significant, because this test will say that the variables are correlated among the population. Obviously, factor analysis technique is applicable when the variables are correlated. Obviously, right? So its value has to be significant. That means p value has to be less than 0.05 for Bartlett test. Then factor analysis technique is applicable. Number one. Number two, KMO test. KMO test is called Kaiser-Meyer-Olkin test. It is used for sampling adequacy. That means KMO test value has to be, its value is always between 0.5 to 1. Remember this value, not 0.05. It has to be between 0.5 to 1. Let's say you apply factor analysis. Your KMO value says that it is 0.35. Do not apply factor analysis. Do not apply factor analysis. It is not applicable to your data. So two tests you have to see. One is Bartlett test. It has to be significant. Let me just repeat. I will not repeat again and again. So Bartlett test value has to be significant. Number one condition. Number two, KMO value has to be between 0.5 to 1. If it is, go for factor analysis. If any one of this is violated, let's say this is coming out to be non-significant, or KMO value is less than 0.5, do not apply factor analysis on your data. It is not applicable to your data, right? Somebody may ask question, then what we should do? You cannot reduce dimensionality in this case. You cannot reduce further the dimensionality of your observation. That is the meaning. So don't ask this question that what should I do? That means further reduction is not possible in that case. Okay. So, and two important tests to look into factor analysis. Then there will be one term which is called commonality. Commonality is, I mean, how much variation one variable share with the rest of the variables is called commonality. For example, if there are 120 variables and commonality of V1 variable is 0.79, that means this variable V1 with shares 79% of the variation with the rest of the 119 variables, right? So that is called commonality. Eigenvalues will be for each factor you will get, and you have to retain only those factors where eigenvalue will be more than 1. Then factor loading will be nothing but correlation between the variables and the factors. Factor loading plot you will get, factor matrix you will get. I will tell you the output. Then the important part is to look for the scree plot. So you will get this type of plot actually, this type of plot which is called scree plot. And on scree plot you will get these factors. So in factor analysis, sometimes they write factor, sometimes they write component. Component and factor, they are one and the same thing actually. And on y-axis you will get eigenvalues. On y-axis you will see eigenvalues. So only those eigenvalues will be important which are more than 1. For example, for this factor, for factor number 1, this is eigenvalue. For factor number 2, this is eigenvalue, and both of them are more than 1. Right? Now what about factor 3? Factor 3 eigenvalue is less than 1. Not important. 4, 5, 6, they are all less than 1. If I drop a line here, so I will not consider these factors. I will consider only. And this is given by scree plot. So with the help of scree plot, who don't know understand value meaning of eigenvalue, you can see which factors are more important, whose eigenvalue is more than 1. That's it. Right? So I will apply now factor analysis technique and show you the result. So let me come up to factor analysis. Practically, I'll give you. So this is factor. Let me open up this file. It is not opening. Open with, let me try from here. Yeah, it's coming up. It's coming up. Okay. So let me give you, these are the variables. There is no concept of dependent and independent variables. And I want to apply factor analysis to this. Generally, factor analysis, I have given you one exercise where there are, I think, 18, 19 variables are there. Here are only 6 just for demo purpose. And you can find out how many factors are important. And I'll tell you how to find out. Okay. Go for the, what are these actually? They are measured on, I think, 7-point scale. Yeah, they are all measured on 7-point scale. And let's look at the nature of the variable. Okay, just look at what is the label of the first one. People go to market to buy toothpaste. Now they want to buy that paste which prevents cavities. That is the variable. People have been asked on 7-point scale whether you want to buy that paste that prevent cavities. So if they give answer, if it is important for them, they will rate this as 7. If it is not important, they will, for example, customer number 1 is rating this as 7, that it is important for me. Or second, it is not important, he is rating this as 1, right? Similarly, second question, I like toothpaste that gives shining teeth. Some people are very, very choosy. They want shining teeth only. Forget about anything else, right? So if they like it, for example, this customer number, let's say 10, they like it very much. So give this as number 6. While customer number 18 is an average, and somebody may be having 2 also. This 2, you don't like this. So this is how the responses have been recorded. And if you see other variables, I like toothpaste that gives shining teeth, should strengthen gums, freshens breath, tooth benefit offered by, so our tooth should not be decayed, and it should be used for attractive teeth. So these are the responses, and I want to apply factor analysis for this. Okay, go to analyze, go to, it's a data reduction, this is a data reduction. Go to factor analysis. Now there is no concept of dependent and independent variables. Mark all of them, put it here, take on this side. Go to descriptives, please. You can note down the steps. You have to do this exercise yourself. Click on to univariate descriptives, coefficient matrix, significance level, and the most important is KMO and Bartlett. It will tell you whether you can apply factor analysis or not. So KMO and Bartlett. And then reproduced matrix. Please remember these are important things for factor analysis to be click on. Anti-image determinants, they are not important. Inverse is not important. Continue. Next is extraction. As you know, by default, the extraction is principal component. There are many methods, but principal component technique is based on orthogonal rotation. Orthogonal rotation means factor 1 will be not be correlated with factor 2. Also look at the scree plot, and only those factors will be extracted whose eigenvalue is more than 1 automatically. This is by default. Continue. Then go to rotation. There are number of rotations in factor analysis. Please note down, the most important rotation is called Varimax. The most important rotation is called Varimax, right? Because it is based on orthogonality principle. And then also click on to loading plots. Continue. Then click on to scores and save this as display factor score coefficient matrix by regression technique, not by Bartlett. These are, of course, you can save, but regression is better. Continue. And in options, what you need to click, if you have certain missing values, you can click onto this option, replace with missing with mean. And then go for sorted by size. Why sorted by size? Otherwise, I mean, if you give this option, then it will be, your variables will be sorted by size, that this variable is most important, this is next one, this is next one. Otherwise, you have to look for the weights, that this is number 3 is important, then after that number 15 is important. It will be a tedious task. But if you sort it by size, it is always easier. Then suppress all those where the correlations are very high or the commonality is low. So we have to suppress. You can leave all those which are below 0.3, let's say. Leave all those which are having less than 30% of the variation sharing with the rest. It has to be more than generally 40%, right? So exclude all of them. That's all. So these are the steps required for factor analysis. Continue, press OK. And here is the result, output. How to read the result? First is simply a mean SD table. You know, for each variable, variable number 1, variable number 2, right? And here is a correlation matrix. Now, if you look at the correlations, some correlations are very high, 0.873, this is 0.858, this is 0.640, right? This is 0.640. So some correlations seems to be high, but don't look for the correlation matrix. Go for KMO and Bartlett. Let's see what happens in KMO and Bartlett. So here is KMO and Bartlett. First of all, I told you that Bartlett test, the significance, this p-value has to be significant. So it is less than 0.05. So first test is applicable. Then KMO test value has to be between 0.5 to 1. And what is its value? 0.66. That means that you can apply factor analysis. We can apply factor analysis, right? And this is a commonality. Commonality means variable 1, how much variation it shares with the rest of the 5 variables is 92.6%. Variable 2 shares how much with the rest of the variables? 72.3%, and so on. These are commonalities. And how many factors have been extracted? You see the result. These are eigenvalues. These are the factors. Factors are also called component. Okay. So here what is the first component eigenvalue or the first factor? 2.7. It's more than 1. And it explains how much variation? 45.5. Right? Factor 2, eigenvalue is again more than 1. How much variation is explained by factor 2? 36.9, 82. I mean 36.9. So total will be, this is cumulative, total will be a 45 plus 36. It's come out to be 82. What about factor 3? It is less than 1. This is less than 1. Eigenvalue less than 1. They will not be. They have to consider. You have to consider only those eigenvalues which are more than 1. So total variation explained by your model will be 82 point, and they will be explained only by these two factors. And here is the scree plot. These are the factors. So if you double click here and in double click, if I mark a line, sorry, this is annotation. I think by mistake I have clicked the annotation. Insert a title, insert annotation. Where is the line? Line is something. I'm unable to find the line option, but anyhow, you can easily see that you see, these are the eigenvalues which are more than 1. How many factors? 1 and 2. Rest of them are not. And then you look for the, don't go for this component matrix. This is not to be seen. It has no meaning. You go for rotated component matrix. You have to see. Then there is a column which is called reproduce correlations. Reproduce means after the F1, F2 have been extracted, what are their correlations? When you include all, they have to be almost 0 because they have to be uncorrelated. You can see this is residual correlation. Was it a residual? Residual means actual value of the correlation minus the predicted value of the correlation. This is residual. So residual has to be small. So model seems to be good. Then this is you need to see rotated component matrix. Don't see the first one. See the second one, rotated component matrix, and they are in order. Now 2 components have been extracted. For example, look at component number 1. 3 variables are there. Variable number 1, variable number 3, variable number 5. And these are their loadings in order because I sorted by size. So the first importance goes to the number 1. So important to buy toothpaste that prevent cavities. Most of the people, they want this, right? Second priority goes to not to V2, but it goes to V3 because its weight is 0.934. Third goes to this. And these 3 factors, they are under 1 component. That means I have to see, then I have to give the name that these 3 factors are really representing what, right? And then there is a second factor also. And these are the loadings for second factor. If you look at the second factor, which is the most important, variable number 6 for attractive teeth. People like attractive teeth. Second one, freshens breath. Third one, shining teeth. Okay. Now let's see how these factors are being built. Look at variable number 1, variable number 3, variable number 5. What these represents? Now read. Important to buy the toothpaste that prevent cavities. That means related to health. Okay. Second one, toothpaste that should strengthen gums. Again related to health. And toothpaste for this is V1, V3 strengthen gums, and V5 is that tooth decay bit benefit offer. I mean, my tooth should not fall. Then this is also health. So these three variables are relating to health. So I can call this component or a factor as health benefit factor. You have to give the name yourself. Please remember, SPSS will not assign any name. You have to choose your name yourself looking at the nature of the variables. So I can call this as health benefit factor. Let's look at the other factor. Attractive teeth, freshens breath, shining teeth. They are all like cosmetic factors. They're all cosmetic factors, or they are from other people's point of view. I can call this as social factor. So either you can call this as cosmetic factor or a social factor, and this is a health benefit factor. And luckily, by chance, this is odd number, V1, V3, V5. By chance, this. So this factor number 1 is by in terms of odd number, and this is by even number. It will never happen, but by chance it has looked like, and easier to remember, that health benefit are in terms of odd numbers, and social benefit are in terms of the even numbers. And if you look at the plot, this is the plot. So these are all even numbers on y-axis. These are all on x-axis. This is factor number 1, factor number 2, and they are explaining about 85% or 82% point something percent of the variation. So this is the entire. And where are these factors actually? These are the factors. What are the linear combination? This is a factors which were under factor V3, V1, V3, and V5. This is a linear combination of that. First one. This is a linear combination of, and if I want to find out the correlation between these two factors, let's see what is the correlation between them. It has to be 0. Correlation, bivariate correlation between this and this. Look at the Karl-Pearson correlation coefficient. It is 0. You see, this is 0. This is what I want. That factors which I have extracted, they should be uncorrelated. So this entire technique is called factor analysis. So I have explained you the factor analysis. Now let me go through the last and the important technique, which is called cluster analysis. Cluster analysis will take around 10 to 15 minutes. And then whatever you will say, I will try to cover. Maybe in next 10 minutes or question answer I will take up. Okay, let's go for cluster analysis. So here is a PPT. You can read this factor analysis PPT later on in detail. And I have given this example here itself. So these are the steps you need to follow, although you have seen practically also. Let's go for cluster analysis. Now, what do you mean by cluster analysis? Is the host available there?
Yes, sir.
Just
Yes,
give me 1 minute. Let me take water.
sure, sir.
अभी कितने participant बैठे हुए हैं? 110. Still good number. Okay. Let's go for cluster analysis. And don't try to learn each and everything today itself. I have given you enough material even for discriminant analysis. Everything, right? And discriminant analysis, maybe I don't think I'll be able to cover because that will take another 20 minutes. But anyhow, look at the cluster analysis. What is a cluster analysis? As the name suggests, we want to form clusters. Let me give you a practical example. Let's say I'm teaching a class, and when the period is over, then 5-6 students together, they move into one direction when there is a lunchtime generally, and 5-6 girls, they move in another direction. 2-3 students, they move in some other direction. And sometimes there is a one person who is totally I mean, moves into another direction. What is this actually? They are basically moves in clusters because they like the qualities of each other. They like the habits of each other. This is called a cluster. That means within a cluster, the items are very much similar. And if you compare this with another cluster, there will be dissimilarity. So what is cluster analysis? Cluster analysis is a technique so that you can combine the items based on their similar characteristics. And for this purpose, I can use cluster analysis. It is very useful. I'll give you one example that if there are customers and you take the opinion of the customers, then I want to see which customers are specifically going after the brand, which customers are specifically going after the price, which customers are specifically going after the cheap quality, and so on. So you can identify those clusters. So this is a very, very important technique. Let me give you the idea. So, I mean, you have to join by hands. If you join by hands, let's say these two are liking each other, and then there is a third one, then they can mix up with the third one. So third one also goes to this cluster. If they don't, then it will go to the another cluster. That is just the idea of this diagram. What is a cluster analysis? In cluster analysis also, there is no technique like independent or dependent variable, just like in factor analysis. So cluster analysis is a class of techniques used to classify your objects or cases into relatively homogeneous groups. What do you mean by homogeneous? That means they are similar to each other, right? So you can read this in detail. Let's say look at the very ideal clustering situation. For example, I have two variables, price and quality. So these are the items which are low on quality, because this is low on quality but high on price. So I can identify. These are medium on quality, medium on price. So this can be clubbed into. But look at this one. This is high on quality but low on price. And look at this one. This is high on quality and high on price. So if you have such type of situation, then it's very easy to identify the clustering, right? But practically the situation will be different. Let's look at a more practical situation. This will be the situation. Make clusters out of this data. And today we will make clusters out of this data. How many clusters can be found? Very important technique. It's very, how you can classify that they belong to one class, they belong to another class, and how many total classes could be there? Whether 4, whether 3, whether 2. So all answer to this is given by cluster analysis. Okay. So there are two types of cluster analysis. One is called the hierarchical clustering. And in hierarchical clustering, we work on a schedule which is called agglomeration schedule. We work on this for hierarchical clustering. Then there is a non-hierarchical clustering. Non-hierarchical clustering is also called k-means clustering. Let me just repeat. There is a hierarchical clustering which works on a schedule which is called agglomeration schedule or agglomerative schedule. And then there is another clustering technique that is called k-means clustering, or this is called non-hierarchical clustering. So we have to use both of them in tandem, one by one, to find out the number of clusters from one into another so that I can get the answer. Okay. So how this techniques works, I'll just explain you the crux. I mean, if there are 2 points, I can compute the distance between 2 points. For example, if one point is 5, another point is 3, right? So that the distance will be 5 minus 3, 2. So, but there will be number of points because there are V1, V2, V3, there are number of points there. Then it is based on a vector notation. And in vector notation, this distance is called Euclidean distance. So first of all, it combines only those vectors together, or only take those distances which are having minimum distance, right? Which are having minimum distance. Let's say item number, variable number 1, variable number, or customer number 1 and customer number 18, their distance is minimum, they like each other, and this distance is 1.2. So they will combine this first. Then one center will be there, one center will be there, and then they look for which one is closer to that center. And if it is closer to that, you combine the third one with them. Then you combine the fourth one if it is closer to them. You combine the fifth one. But let's say sixth one is very far away. It is crossing some threshold point. Threshold point will be there, and it is crossing, then it will go to the next cluster. So it will, this is how the clustering techniques goes. So it is based on a method of centroid. So I'll not go into details, but I have given you, and you will not have to read all this. So there is a very important picture, just like we have a scree plot that will give you idea about how many factors will be expected. Similarly, dendrogram will give you idea how many clusters can be there in your data. So look at the dendrogram and find out the number of clusters. Okay. And then put those number of clusters into k-means clustering method, and you will get the answer that these are in this cluster, these are in this cluster. Okay. There are certain statistics which are associated with the cluster analysis. For example, one of them will be Euclidean distance. Euclidean distance we need to compute. It will be computed by SPSS. And let's say, look at this data. So this data is again on 7-point scale. There are certain variables. We will make clusters out of this data, right? And you will get this type of matrix. Then how many clusters can be there and which item will go to which clusters, we will clarify. Okay. So with this background, you can go through this. There are number of things, other things. I mean, what is the difference between ANOVA, the regression and discriminant analysis? It is given in this slide. You can go through. Then there is a discriminant analysis slide also. And I have given you example so that you can do it yourself for discriminant analysis. Data is already with you, right? So with this background, let me come to the practical demo of the cluster analysis. Okay. Cluster analysis. Let me go through this file. Okay. See, experiment is like this. There are customers who are visiting the malls. There are 20 customers. We want to classify that which are the customers which are having similar properties and those who are having dissimilar properties. We want to make clusters out of this. And there are certain features, and features are like this. That whenever people go to mall or they go for shopping, the first question have been asked that shopping is a fun. For some people, it is fun. Those who are very rich, it's just a fun for them. But for mediocre people or low income people, it may not be, right? So if it is fun for them, they will rate this as 7. If it is not, then they will rate this as 1. Then shopping is bad for budget. I think wife should also ask the husband whether it is good or bad. I combine shopping with eating. Again, this response has been taken. Sometimes people go to mall. Whenever they say, "I'll take dinner on. We will take dinner there only with the family." They take such type of decisions. I try to get the best buys when shopping, right? I don't care about shopping. You can save a lot of money by comparing prices. So all these responses has been captured in this data file, and this is the data file. Even in terms of, I think a 7-point scale has been used. So our problem is to make clusters out of this data. Okay. So let me make clusters out of this data. So you can note down the steps, and I have given you a cluster exercise. Please try to do that exercise. Go to analysis and go to, let's say, this classify, because it's a classification technique. And in classification techniques, first of all, you have to go for hierarchical clustering. Then we will use k-means clustering. First, please remember, we have to use both of them, k-means as well as hierarchical. From hierarchical clustering, you will come to know how many clusters can be formed, number one. Then once the clusters are known, then k is known. That's why it is called k. Then put the value of k there. Suppose 5 clusters are known to you, then k will be 5. So I'll use this hierarchical clustering first. There is no concept of dependent and independent, but case number should not be included. Case number should not be there because it's a case number, right? It's not a response. So take all these variables here. Then which are the statistics to be computed? Go to statistics. Now it is asking number of clusters. I don't know how many clusters are there. So just leave it blank. Go to plots. And the most important plot is dendrogram. Dendrogram will give you the number of clusters that can be formed. Okay. Automatically by the data. Continue. Then method. Now, which method I told you? Euclidean distance method. So it's a squared Euclidean distance, and then click on to Wald's method. Please remember, the best method is Wald's method because it is based on minimum variation principle, that within a particular cluster, variation of the items will be less. And if you compare with the other cluster, they will be more. So Wald's method is based on minimum variance principle. Then squared Euclidean distance will be computed. Okay, continue. And you will not have to, I think, save anything right here. Okay, that's it. For hierarchical clustering, go to OK. Now, if you press OK, you will see, you will have to see this table. This is very, very important. Please look at this table, which is called Wald's table or Wald's linkage table, based on the agglomeration schedule, which is for hierarchical clustering. Now, first of all, it combines those customers. There are 20 customers whose distance is very, very small, and Euclidean distance is computed, and this is coefficient is called a Euclidean distance. For example, which customers are having closer, I mean similar properties. For example, what is first one? First one is 14 and 16. 14 and 16. And what is their distance is very small, 1. That means, first of all, customer number 14, customer number 16 will be combined. This is what it means. Because this is coefficient you can read as a distance, right? And then leave everything, go to the last column. What it says, next customer that will come will be, go to step number 6. Next stage will be 6. Don't go for second line now. Go for stage number 6, step number 6. Now, in step number 6, if you see, out of 14 and 16, one of the number will appear. Either 16 will appear or 14 will appear. Look at this. In 6, which one is appearing? 14 is appearing. Out of the 16 and 14, one of the number will appear. 14 is appearing. Then 14 is here. Then the next that can be combined with 14 and 16 will be 10. This is what it means. 14 is there, so next item will be 10. Okay. So now you have 3 items in one cluster. 14, 16 and 10. All right. Then go to the next stage. What is the next stage? It says go to 9. Go to 9 now. Go to 9. And out of 10 and 14, one of the number will appear. See which number is appearing? 10. And the next item that will be clubbed with this cluster will be 4. Then go to 12. This is how the table is being read. Hopefully this makes sense to you. And look at the dendrogram. This is a very confusing picture, which is called isosceles diagram. Just don't go for this diagram. This is a very important picture. And it will give you, you see 14, 16, 10 and 4. Next will be 19, next will be 18. Now you have to identify gaps. Where is the gap? Generally we take, sometimes depending upon the number of observations, sometimes we take 5, sometimes take gap of 10. But if I, let's say, look at gap of 5, this is the first gap which is appearing here. This first gap on x-axis you see. After that there is no. Okay. Then there is one gap here. There is one gap here. One gap here. Okay. Now, how these have been clubbed based on that table, 14, 16, 10. So these are the items which goes to one cluster. Which one? 14, 16, 10, 4, 19, 18. And then 2, 13, 5, 11, 9, 20. They will go to the second cluster. And these are goes to the third cluster. So basically look at the gaps. One gap, 2 gap. So one cluster, second cluster, 3. So basically from the dendrogram, I can see there may be 3 clusters, roughly, right? There may be 3 clusters. So that is the role of the dendrogram and that table. Now I'll go for k-means clustering. Now I know the number that there will be 3 clusters. Go for analysis. Go for classify. Go for k-means cluster. Now in k-means cluster, you see, take the variables on this side again. Now you have to count that which case goes to which one. Case number label by this. Label cases by this. And number of clusters. How many clusters are there? So it is written 2, we have to make 3 because number was 3, right? And then go to, you cannot apply k-means directly because it will ask you how many clusters. You don't know how many clusters. So first go to hierarchical, then go to k-means. Then go to save cluster membership. We need to say cluster distance, which are the distances. I mean, that table, if you want to save, you can save that table, but we don't require. In options, what we need, ANOVA table you can have. That's it. Okay. Press OK, and you have this. So it says these are the clusters for each mean. This is not important. The most important is ANOVA here, that in each cluster there are items and they are significantly. For example, the first item, if it differs in all the 3 clusters, so degrees of freedom is 2. So first item, shopping is fun. So there is a significant difference. Second variable, significant difference. So all they are significant. They differ, I mean, they have to differ because they are belonging to the different clusters. They are belonging to the different clusters. Now it says, how many items are in cluster 1? 6. How many items are in cluster 2? 6. And how many in cluster 3? 8. And altogether 20. Now I want to see which are the customers which goes to, which are these 6 customers? Which are these 6 customers? Which are these? Go to the table. We have already saved. This is a cluster membership. You see? This is a cluster membership. And wherever there is 1, you see 1 is here. Wherever there is 1, that will go, I mean, if you see cluster number 1, I mean customer number 4, then customer number 10, we have already seen 4, 10, and then I think 16 was there, 16, next was something. So all they are going to cluster number 1. Cluster number 2, see this customer is going to cluster number 2, this is going to cluster number, and you can identify. And you can also make a graph of this, how many people in each, so you can draw a pie diagram. Let's say draw a pie diagram for a group of cases and take this cluster membership here and make a pie diagram. So this is your pie diagram. And you can represent in a nice way, show data levels. So these are the percentages, you see. So that means 40% of the people, they belong to cluster number 3. Then 30% they goes to cluster number 2 and 30% here. So you can make a nice picture, present your results. So this is all about the cluster analysis. So I have complete the factor analysis as well as discriminant analysis. I think many of the participants, one is left is ANCOVA, MANOVA, and the third one is discriminant analysis. Anyhow, I am conducting another workshop. If you are left with the, I'm conducting another workshop from 24th to 26th, and it is for those people who want to join that workshop. I'll send you the information because you are in the WhatsApp group, and it is finalized today only, and it was conducted by some Pune Institute. I don't remember the name of the Pune Institute, and it will be held on 24th, 25th, and 26th of this month. And where there are discriminant analysis, MANOVA and COVA, etc., they are mentioned there, so they will be covered. But they are not covering some other topics, so that's why those will be covered. So sorry for that because time is up, and moreover some questions will be there. I have to take up some questions. Don't worry at all. It is not important to learn all the techniques in statistics. These are the most important techniques which you have learned. In fact, okay, ROC curve, logistic regression, nobody teaches these techniques. And also the cluster analysis, factor analysis, these are the most important techniques. So if you have any questions, please, you are most welcome to ask me.
Yes, sir, we have many questions lined up.
Yes, please.
So shall we begin?
Yes, why not?
So first question is, can you cite an example in medicine for cluster analysis? 2 or 3 participants requested this question. an example from medical field, medical sciences for cluster analysis.
Cluster analysis,
Yes.
medical field. Okay.
Mhm.
You see, let's say there are, randomized control trial, right? Yeah, there is a technique which is called randomized control trial. In randomized, there is a single blind trial, double blind trial, and people are giving, let's say, medicines, and they are having groups of patients. But neither the doctor knows which medicine has been given, neither the statistics person knows which medicine has been given. It's called double blind, and it has been controlled by some third party, right?
Yes.
Now you have the responses. Let's say responses are in terms of betterment of a, or you can say what is the recovery time of a patient. Now you want to make, you have a recovery time of the patients based on medicine number 1, medicine number 2, medicine number 3, right? And certain features of the like blood pressure, cholesterol, height, weight, etc. You can have, I mean, you can ask the which are the features to be important for such kind of disease. You can put them into, and based on all these features, now you can make clusters, and you can see if your clusters identify that these patients are of in the same group, and they have been given this type of treatment, and these are in another group, they have been given second type of treatment. I mean, you can identify the treatment that these are the similar which are having this type of treatment they have received, the second and the third. So these are the examples in medical sciences, how you can club into groups during the
Okay.
randomized control trial procedures.
Okay, sir. Okay. So next question is, if in agglomeration schedule, if next stage is 0, what to do? Does that mean one cluster is complete?
No. Next stage will never be 0. Next stage will never be 0, because it will be coming out of 1 to 20 only. It will rotate. It will come 1 to 20. There are 20 customers, so it will come. Either it will go to 19, maximum it can go to 19, right? The 19 items have to be clubbed. It cannot be 0. It can never be.
Yes, sir. Yeah. Okay. So next question, how do we come to know what is common in a cluster?
They have been combined on the basis of the similar characteristics, wherever the distances are very, very low. For example, there is one patient whose BMI is 23. There is another patient whose BMI is 23.2. There is another patient whose BMI is 30. And there is another patient whose BMI is 22.9. Now, these three patients whose BMI is closer, 22.9, 23.2, 23.3, they are very close. They will be put into one cluster, while 30 will go to the another cluster. This is how the clustering techniques works.
Okay.
So items goes which are having similar, they will go to one cluster.
Okay, sir. So next question, what should be the design effect for such cluster studies if we got similar responses from the participants? How we may compare it with Paul's method?
Now, you see, design effect is not computed for cluster analysis. Design effect is not computed for cluster analysis. They are for randomized control trials. In that case, we generally do, but not in case of cluster analysis, unless you are using some stratification techniques, which is beyond the level of this workshop. There are certain stratification techniques using cluster analysis where we use effect size, but I have not covered that part. In fact, that will take another maybe 1 hour to explain you what do you mean by effect size. Yes.
Okay, so next question has just come up. How can we find the mean and standard deviation of a variable as BMI of the respective clusters?
See, there is a tick option. You find out the number of items. Let's say there are 6 items in one cluster, 6 items in another, and 8 in the third. Then automatically the one table has appeared. That table has given you the mean and standard deviation. The table was there, in fact. I have not, that table was not important, so I have not given the interpretation, but the table is there in the output itself.
Okay.
It is there, mean and standard deviation table for each cluster.
Okay. So how to calculate sample size for factor analysis and cluster analysis?
You will not have to calculate any sample size for factor analysis, neither for cluster analysis. For cluster analysis, I mean, there is a important test. I mean, you can just look at the dendrogram And if there are no gaps in between, that means you cannot form clusters out of this data. You cannot form, right? That is the simple test to look into. Another is for factor analysis also. I mean, factors are extracted, or two important tests I have already told you. One is KMO, another is Bartlett. So just look at KMO value, just look at Bartlett test. Even if your sample size of 2830, doesn't matter. Even if your sample size is 2000, doesn't matter. Sometimes with sample size of 2000 even, your KMO value will be less than 0.5. That means even for this sample size, you cannot apply factor analysis. So there is no question of, you have to just look for KMO value and Bartlett test. If they are satisfied, you can go ahead with the factor analysis. Otherwise, you cannot.
Okay, sir. Next question is, how do we see unstandardized and standardized odds ratio in logistic regression?
You see, there is a comparison. I mean, if you want to form an equation of the logistic regression, like y is equal to beta naught, we always use unstandardized coefficients. Please remember, we always use unstandardized coefficients. And standardized coefficients make sense whenever we want to compare them, that which one is better, which one is, for example, age is better or cholesterol is better. Then you look at the standardized coefficients. The coefficients which is having higher value will be better. The coefficient, standardized coefficients, and which is having lower value will be having less importance. So standardized coefficients are used for comparison, while unstandardized coefficients are used to form an equation.
Okay. Okay, so next is, can we use cluster analysis for descriptive studies?
You can use, if your problem is to make clusters out of the data and certain features you observe, because in descriptive analysis you observe certain features and you want to classify, then you can use definitely.
Okay. Okay, next question, the example of logistic regression taken up for discussion. What test is the ROC curve talking about? There were variables used and no tests. So please explain that.
Actually, the surgery was performed in that case, and we have seen that there was no effect of surgery, but there was an effect of history of glaucoma. Actually, you have seen that history of glaucoma was coming out to be significant. And we have that after the surgery, whether the patient has developed glaucoma or not. So it was based on age. It was also based on history of glaucoma. Now, which one of this is categorical variable? The history of glaucoma was a categorical variable, and then there was a response variable. So it was basically looking into the odds ratio for history of glaucoma. And if I have not shown you the odds ratio, but odds ratio was computed there. You just look at the odds ratio, that the people who are having no history and those who are history, let's say their odds ratio is 1.5. That means those people who are not having history and those who are having history, they are having 1.5%, 1.5 times higher the chances of developing the post-PK glaucoma. So it is based on the odds ratio. And this odds ratio also classify that whether our test is good or bad, right? So if you see, there was a classification that out of this has been correctly classified, this has been non-classified, and the entire table was based on the sensitivity and specificity. For that, the ROC curve was there.
Okay. So next question, in SPSS, is there any way to find 95% confidence interval for sensitivity, specificity, NPV, PPV, etc.?
I haven't checked that one, but I think it should be. It always gives the 95% confidence interval for odds ratio. I think if you look at the ROC curve again, maybe you can check. I think it is there. I think whenever you have area under the curve, I am 100% sure. One of my paper is also there in class 1, where I have computed the confidence intervals for area. For area, there is a confidence interval. I am 100% sure. But whether sensitivity or specificity, there is a confidence interval or not, I'm not sure. Not sure. You have to check the output. Not very sure. I don't remember now. Yeah.
Sure. What is the difference between generalized estimation equation by linear regression versus MLR? And what if constants are significant, whether to comment or not?
You see, generally simple linear regression or multiple regression techniques, they work on a principle of method of least squares. method of least squares consists of minimizing the error sum of squares. So it computes the errors, then square those errors, and then take some differentiation and minimize those errors. So maximum, it is simple regression, multiple regression techniques. The coefficients are estimated based on the principle, mathematical principle that is called method of least squares. But in logistic regression, there is a categorical variable. Dependent variable is also categorical. Independent may be categorical or continuous. So this least square method we cannot apply in logistic regression. Then there is another method. If there are categorical methods, then there is another method which is called the maximum likelihood technique. It's very complicated technique. It's not so easy, but I mean, you have to go through a lot of statistical equations or mathematical equations to solve this. But there is a technique which can solve the equations, and these equations can be solved with the help of maximum likelihood. So maximum likelihood technique is applicable in many situations. For example, in discriminant analysis, it is also used. In discriminant analysis, it is used because there is a factor also. I mean, dichotomous variable, dependent variable is dichotomous. In logistic regression, it is used. And in many other applications, it is also used. So it's a complicated technique to estimate the parameters, and generally used whenever there is a categorical variables.
Okay. Okay, so next is, can we apply PCA for mixed data having both categorical and continuous data?
No, you cannot apply PCA for that. Generally, either it has to be uniform, either on a 5-point scale, or maybe on a 7-point scale, or a 10-point scale, or it could be measurements like height, weight, BMI, cholesterol level, blood pressure, all this. You can apply principal component, not on the different scales. For example, one variable is on 5-point scale, another is yes, no. You cannot apply factor analysis. Then there is another technique. Let's say all your variables are in terms of yes and no, and you want to apply principal component. You can apply for this data only if 1 and 0 are there, but that is not called the state of a principal component analysis. That is called the name I'm just forgetting. It's called categorical principal component analysis. Categorical principal component analysis. That is only for categorical data, specifically only for categorical data. Or sometimes I think, if I'm not, I think it is also called sometimes the component PCA or something like that. Component PCA, I think component PCA.
Okay.
Exact name, you can Google search and find out.
Sure. Okay, sir. So one more question. If communality of some variables are less than 1, should we remove those variables?
No. Commonality is always between 0 and 1. So if any variable is having commonality less than 0.4 or 0.3, you can remove that variable. That is not important then. It can never be more than 1. Commonality always lie between 0 and 1. So you can remove those variables which are where the commonality is less than, let's say, 0.3. Why? Because with the rest of the variables, it is not sharing any variation. Let's say it is only 0.1. Then only 10% of the variation is being shared. And factor analysis technique is used whenever more sharing is there. So it is sharing only 10%. You can remove that right in the beginning.
Okay. Okay, sir. How to save predictive probability for ROC?
There is a option there. You go to option and there is a save option. You click, in save option, you go to save, and predicted probability option is there. Just click that and it will save.
Okay, sir. So what are hat values and why are they used?
Hat values?
Yes, sir. Somebody had put that question.
I have also put in hat. Because during the lockdown, I have removed my hairs.
Okay.
See, hat value means, in statistics, hat value means estimated value, estimated from the data. When the values are being estimated from the data, then it is called beta 1 hat, beta 2 hat, beta 3 hat. That means they are estimated values from the data. They have been estimated and put in the equation. They are known values then. This is what we mean by hat.
Okay. So one more question. Which regression to use if dependent variable is ordinal?
Ordinal regression. Then there is a ordinal regression which cannot be covered in this, which was not covered in this. And if your dependent variable is having number of, let's say number of people who die from air pollution, number, the dependent variable is number. Let's say 25 people die from air pollution on this day, and 30 people die on this day, and 35 people die on this day. So it's a number. It's not a measurement. Neither it is a categorical variable. For that we use Poisson regression. And if it is 1 to 3 form, we use ordinal regression.
Okay. Okay, sir. Please explain the term testing of data. Testing of data.
Testing of data means you are testing the normality basically.
Yeah.
You are testing the normality, checking the normality of the data for further treatment, whether to apply parametric test or to apply non-parametric test. So I have given you methods to check the normality: Spirovilk test, KS test, then histogram, box plot, QQ plot. Number of methods are there to check the normality. Yes,
Okay.
please.
So out of all these output results, what all should we have to show in our final results for factor analysis?
The most important for factor analysis is a scree plot you can show, right, for factor analysis. And
Okay.
that table which was having eigenvalue and all that, total variation explained, that is the most important table to be shown. Where there was a eigenvalue, component 1, component 2, that was the most important table. You have to show that table and the scree plot. These are the two important things. And KMO and Bartlett. KMO and Bartlett. The rest of the tables are not important.
Okay.
Or of course, for you can also show the rotated component matrix. Rotated component matrix, and there was a discriminant score matrix also. These two are also important. You can show them.
Okay, sir. Also, somebody is requesting for just the concept of discriminant analysis.
I can give you the concept of discriminant analysis in 1 minute. Okay. See, what is a discriminant analysis? Discriminant analysis means basically it's a classification technique. Whether you can classify an individual to this category or this category. Let me give you a very simple example of, let's say from medical field itself. Let's say in ancient times, people used to find, there are different type of skulls, and skulls have been found now in Amritsar, and we are doing research on that. I'm involved with that project. So number of skulls are there, right? And number of skulls, I think there are I think of maybe 1920s or before that. Now we want to classify whether it is a male skull or it is a female skull. I don't have any idea whether it is a male skull or it is a female skull, right? Now there are certain parameters for skull. There are bones in our skull, and 15 main bones are there. I can call these as, let's say, width is there, length is there, diameter is there. So many different type of parameters are there. Let's say these are 15 numbers. I call this as x1, x2, x15. Okay. And then there is a male and female. Now, in the same area, people are dying. And initially now they are in the mortuary or wherever they are. Initially, you know that in the same area, obviously these people, they almost belong to the same area, right? So obviously maybe in 100 kilometers within themselves. That's why the skulls are there. Okay. Now within that area, what you do, you take a sample. Let's say I take a sample of 100 people. Some of them are female. Let's say 50 are male, 50 are female. Okay. So initially I will be having a dependent variable. Let's say I write male as 1, female as 2. So I will be having one column. And then I will be having the measurement of all these skulls. So 50 values are x1, x2, x15. They are available for male. They are also available for female. Okay. Now based on this data of 100, just like we apply regression analysis, we apply discriminant analysis. And you will get discriminant score equation. You will get discriminant score equation. And along with discriminant score equation, that will involve which type of variable? X1, X2, X15 variables. They will be involved in discriminant score. This is called DS. And simultaneously, you will also get two centroid. One centroid is called C1 for male, another centroid C2 for female. They will be known to you from the given data. Now there is an unknown skull. I want to identify whether it is a male or female. You measure all the 15. You can measure all the 15 measurements. Put in this equation, x1, x2, x15. You will get the discriminant score. So your discriminant score will be computed. Now you see whether this score is closer to the centroid of male, then it will be classified as male skull. If it is closer to the female skull, then this will be classified as female skull. So anthropology, people from anthropology, people from forensic science, people from anatomy, they are using this technique of discriminant. How to classify an individual to a group, whether this will go to male or this will go to female. So that is called the discriminant analysis.
Okay, sir. One more question. So, in a study of testing different type of errors, whether present or not, in different phases, where the population is different each time, what type of test to be used?
Can you just repeat the question?
Yes, sir. In a study of testing different types of errors, whether they are present or not,
See,
in different phases.
First of all, the question is wrong.
Yes,
actually. We are not testing errors. We are not testing errors. So there is no question actually like this. We are not testing errors. I have never told you test errors. Of course, errors are tested, but they are tested for some other purpose, not from the parametric and non-parametric point of view. They are testing in time series. They are testing in Durbin-Watson test. So there are, I mean, this question is not correct in itself. Yeah.
Okay, so one more question. Adjusted and crude odds ratio, how to compute it? Please explain with an example, please.
Adjusted?
Adjusted and crude odds, odds ratio.
Okay. Because I'll have to open again the
Actually,
SPSS. Time is less.
Shall I interrupt, sir?
Yeah.
Yeah, no. Actually, for you, sir, also, if time is not sufficient, I think we can start with the
let me just answer the last question. Just
Okay,
answer the last
sir.
question.
Okay.
Yes, sir. These are just the last question.
Last question. Okay.
Yes.
You see, adjusted, for example, I want to make an adjustment for age or adjustment for gender. This is an exercise to you. You go to logistic regression. There is a dependent variable and there are independent variables. Then there is a category which is called next. You click on to next and then put the adjusted variable there. Let's say age or gender, and then carry out the analysis. So it will be automatically adjusted. Your analysis will be adjusted for age or gender. Yes,
okay.
please.
Okay, sir. So we wrap up with the question and answer, and I hand over to Dasi sir for the word of thanks and to conclude the session.
Yes, please.
Chitra ma'am.
Yes, Dasi sir is going to conclude. Hello? Yeah.
Yes, good evening, everyone.
Your
It's my
voice is
privilege
breaking.
to have been. Hello.
Voice
Hello,
is
sir.
breaking. Yes.
Hello. Hello. Good evening, everyone. It's my privilege to have been asked to put the vote of thanks on this occasion on the behalf of Department of Community Medicine and Research Cell, MGM Medical College, Aurangabad. Firstly, I am thankful to MGM management for permitting and guiding us to organize the workshop. I am thankful to our beloved Dean, Dr. Rajendra Bohra sir, who is standing behind.
Okay. I think voice is still breaking. Yes, please. But thank you very much. I can understand what you have said. So thanks a lot.
Any other participants who would like to say something?
Yeah, I mean you can
Just
have
this
their
is
have
because
their videos on. I think maybe for 5 minutes.
Yeah.
Then we can close. Just have their videos on.
Dr. Surve, can you please make all of them visible?
And you can unmute. Whosoever raised hand,
Hello.
they can Yes, please.
Yeah.
Hello.
Yes, please.
Uh,
Voice is
please
breaking.
raise your hands. Those who want to speak, can you please raise your hands so that we'll unmute you?
Hello, ma'am.
Yes, please. There is a connectivity issue. Next one.
Hello. Hello, sir.
Yes, yes.
Hello. Hello, sir. Can I audible?
Hello, Dr. Dase, sir. Hello.
Hello, ma'am.
Yeah, your voice was
Hello.
breaking. So, sir says that he has this.
No, sir,
Hello.
no question. Just want to say thanks to you, sir,
No
for
problem.
accepting
Thank
our
you.
invitation and yeah, available to guide us. Thank you for that, sir.
Right.
Hello.
Ji,
Hello,
ji
Hello.
sir.
bataiye.
No, can I audible?
Yes, yes. Now you are. When
Sorry,
picture
sir.
is there,
Sorry.
then you are audible.
Okay, okay. Sorry, sir. Good evening, everyone. It's my privilege to have been asked to propose a vote of thanks on this occasion. Department of Community Medicine and Research Cell, MGM Medical College, Aurangabad. Firstly, I am thankful to MGM management for permitting and guiding us to organize this workshop. I am thankful to our beloved Dean, Dr. Rajendra Bora, sir, who is standing behind us in each and every activity patiently and guiding us to make things happen. Knowledge of SPSS and multivariate analysis. Thank you so much, sir.
Thank you.
Dear participants, thank you for participating in the workshop. We hope that you found the workshop informative and worthwhile. We look forward to seeing you at the next event. We truly appreciate your support. I am thankful to Dr. Shobha Salve ma'am, Professor and Head Department of Community Medicine, for inspiring us to conduct this workshop. I would like to thank all organizing team of workshop for their sincere efforts, especially Dr. Swati Mahajan ma'am, Dr. Sangeeta Achitre madam, Dr. Rahul Surve sir, Dr. Taide sir, Dr.
Again, there is an issue of connectivity. Oh, okay.
Hello.
Yeah. Something.
Hello, sir. Am I
Yeah,
audible, sir?
Yes, yes. Dr. Vivek.
Yes, sir. Yes, sir.
Yes, yes, please.
Sir, thank you so much for this workshop. I would like to congratulate the whole coordinators of this workshop. So actually, this is my second workshop. The earlier workshop which I have attended, that was on structural equation modeling.
Oh, that's great.
Yes, sir. And that was the workshop which motivated me to attend this one. Actually,
Good.
earlier, I'm a student of non-medical background. So earlier I was feeling a little different, or I can say there was a dilemma whether to attend this or not, because I was feeling as there would be a lot of technical jargon which is generally used in medical fraternity. But thank God, when I attend this, so the things were quite good. And this research methodology workshop is quite good. And I would like to attend other workshops also.
Thank you. Thank you. I would like to say something
Thank you
because
so much, sir.
you are from non-medical. I am also from non-medical.
Okay.
But later on, what I did actually, so after doing PhD, I entered into a field which is called biostatistics. And biostatistics involves many of the, I mean, I have to go through all the genetics and all these things together, and bioinformatics. So I have done many techniques over there. I was working with NIH, which is called National Institute of Health in US, which is you. So NIH is, I think everybody knows NIH in US. 99% of the universities of US, they get funding from NIH. So it's a very important institute where I learned many techniques. And after that, written a book for medical doctors. And I was appointed as an advisor by the International Medical Olympiad in Greece last year. And after that, one of the person who is there, Professor Grammaticos, he requested me that people are making mistakes in statistics. Why not to write a book? Then I have written a book. I think I have shared with you that on medical statistics. That book is very important, but that is some involving some advanced techniques like meta-analysis, bioinformatics, and how to look into the genetic association and so on. So that is little bit, I mean, not at this level, but what I'm trying to say that even if you are a non-medical person, you can learn medical terminology and still you can excel in this field. That's not very difficult.
Yes, sir. So thank you so very much for this workshop.
Hello.
जी, somebody else is.
Hello.
Yes, please.
Hello.
Yes, yes, सुन रहे हैं आप, बोलिए.
Yeah. Good evening, Sharma sir. It was very excellent workshop. I like to thank you from bottom of my heart that it was really some of the concepts which I could not touch in statistics. I'm into biostatistics. Basically, I'm MD PSM and into biostatistics by interest. But some of the topics which you have covered today, factor analysis, discriminant analysis, I've never thought of this. So I was just waiting for all those topics. So thank you for creating interest in that. It will boost up further learning. Now, one another request, sir. Now we already have your contact number with us due to WhatsApp group. And I think at least I will be disturbing you a lot. So I hope, sir, you will not mind.
No, I never mind, but only thing is time constraints are there because I'm having already 6 projects with me, WHO, ICMR, and 6 students are doing, 4 students are right now doing PhD with me. So sometimes if I am delay in replying, please bear with me.
No problem, sir. We will just drop our message on WhatsApp. Whenever you find time, you can reply, sir. No problem.
And moreover, one request, sometimes people take advantage. I mean, they send the data to me, please analyze. You see, I don't have this much of time. I can only guide you. But rest of the things you have to do.
Definitely. It will be all conceptual questions and our difficulties where we are stuck up. Thank you very much, sir.
Madam Shobha, Professor Shobha Madam, अगर आपकी इजाजत हो तो एक story सुना दूं. अभी भी more than 60 participants are there.
जी सर. Yes sir.
चलिए एक छोटी सी स्टोरी सुनिए. एक बार एक सेल्समैन था और वो अपने ऑफिस में, हिंदी में सुनाता हूँ, हिंदी में स्टोरी सुनाने का सबको आती है ना हिंदी तो?
जी सर.
Everybody understand हिंदी, right?
Yes sir.
Yes sir.
तो एक सेल्समैन था और उसको उसके बॉस ने बहुत काम दिया, तो वो काम लेके घर पे आ गया. तो काम लेके घर पे आ गया तो उसका एक 5 साल का बेटा था, 5 to 6 years old. वो अपना लैपटॉप पे और अपना रिटर्न में कुछ काम कर रहा था, लेकिन वो बार बार उसको खींचे जा रहा था, काम नहीं करने दे रहा था. तो वो परेशान हो गया. वो कहते हैं कि अब मैं क्या करूं, ये तो मेरे को सुबह तक करके देना, ये तो बिल्कुल काम नहीं. घंटा हो गया, 1.5 घंटा हो गया, वो काबू नहीं आया. फिर उसके माइंड में एक दिमाग में बात आई, शाम हो गई थी, तो उसने क्या किया कि देखा कि इवनिंग न्यूज़पेपर में एक वर्ड का मैप है, एक वर्ड का मैप दिया हुआ था. तो उसने वो वर्ड का मैप उठाया और बेटे को बुलाया जो 5-6 साल का था. कहता बेटा ये क्या है? कहता वर्ड मैप. तो उसने क्या किया कि उसके सामने उसको टुकड़े टुकड़े कर दिया और उसको एक टेप दे दी और उसको बोला कि आप इसको जोड़ के दिखाओ इसी तरह से जैसे ये है. वो सेल्समैन का ये आइडिया था कि भाई इसको तो बहुत 2-3 घंटे लग जाएंगे जब तक मेरा काम खत्म हो जाएगा. लेकिन जस्ट आफ्टर 3-4 मिनट्स उसने वो सारा जोड़ के और अपने पापा की टेबल के ऊपर रख दिया. कहता ये लीजिए. वो हैरान, कहते ये तूने कैसे कर दिया, ये हाउ इट इज पॉसिबल कि तूने वर्ड का मैप ही इधर जोड़ दिया. तो कहता है कि पापा जी इसको उल्टा करो. तो जब उसने उल्टा किया तो क्या देखता है कि वहां पे एक आदमी की फोटो थी. कहता है मैं ये आदमियों को जोड़ता चला गया, इसकी बाजू जोड़ी, ये सब कुछ जोड़ता चला गया और ये दुनिया जुड़ती चली गई. वही काम हमने इस वर्कशॉप में किया. हम तो सिर्फ लोगों को जोड़ने की कोशिश करते हैं. पिछले लॉकडाउन जब से शुरू हुआ है, मेरे ख्याल से मैंने 8,000 लोगों को ट्रेनिंग दे चुका हूँ अभी तक. 8,000 people. In one of the workshop there were 4,300 people in one of them. तो वही काम जोड़ने का हम लोग करते हैं.
Congratulations sir for that.
Thank you.
So just one thing I like to add for team MGM Medical College Aurangabad. Thank you very much to entire team. I'll not take any name here because there are so many people and I'll forget something. So I don't want anybody to get hurt. But thank you to entire team. Very nicely organized workshop. And just one request, the group which we have created, if we can continue it with Sharma sir, definitely if sir is okay for that, then we can have same group to continue our discussion in statistics. Thank you very much.
Thank you. So anything else? एक शेयर सुना दिया जाए आपको? मैंने कहा एक शेयर सुना दिया जाए?
Yes sir, yes sir, बिल्कुल sir.
चलिए शेयर सुन लीजिए. मेरे पास तो मैं खुद अपने शेयर खुद लिखता हूँ.
तो ग्रुप में शेयर कीजिए सर।
नहीं मैं ग्रुप में शेयर नहीं करता क्योंकि वो लोग फिर ये सोचेंगे कि ये स्टैटिस्टिशियन है कि कोई दूसरा आदमी है। हर कदम पे इम्तिहान लेती है जिंदगी। आप देखिए शुरू से बचपन से जब से पैदा हुआ इम्तिहान ही ले रही है जिंदगी कि नहीं ले रही?
Yes, sir.
हर कदम पे इम्तिहान लेती है जिंदगी, हर वक्त नए सदमे देती है जिंदगी। हम जिंदगी से शिकवा कैसे करें, आप जैसे लोग भी तो देती है जिंदगी, आप जैसे।
वेरी गुड सर।
थैंक यू।
कहते हैं ना कि जिंदगी किसी की मोहताज नहीं होती, जिंदगी किसी की मोहताज नहीं होती, दोस्ती सिर्फ जज्बात नहीं होती, कुछ तो ख्याल आया होगा खुदा को, वरना आप सबकी इस वर्कशॉप में हमसे मुलाकात नहीं होती।
क्या बात है, क्या बात है सर।
तो मेरे पास तो अनलिमिटेड स्टॉक है, आप सारी रात भी बैठ सकते हैं। कहता हर राहें आसान लगती हैं, हर राहें आसान लगती हैं, मुश्किल जो भी आए वो भी मुस्कान लगती हैं। तो हम तो कभी देखते ही नहीं रुकावट को, हम तो देखते ही नहीं किसी चीज।
So we request Dr. Shobha Salve ma'am to conclude, ma'am.
Yes, please.
Yeah, I request Dr. Das is there. I'll conclude it. Das is sir.
Yes,
Yeah,
yes.
he's there.
Good evening, everyone. It's my privilege. Once again, I am vote of thanks, ma'am. I'm giving once again vote of thanks.
नहीं आप
Is it okay?
नहीं हो गया, आपका हो गया vote of thanks.
Okay, sir.
Yeah. Anything
Once
else you
again,
want to?
I am
Anything else you want to say? Add. Yes, please. Sorry, क्योंकि टाइम इतना बचा नहीं कि जो बाकी एक दो टॉपिक बच गया वो कवर नहीं कर पाए, बट चलिए कोई बात नहीं। आई एम होल्डिंग अनदर वर्कशॉप, मैं उसका आपको भेज दूंगा। It is just finalized today only, 24th, 25th and 26th, then and Kova Manova will be covered there. And
No,
some
sir, you are
additional
trying to
topics
cover
are
all
also.
the topics. You are trying to cover all the topics, but it's not possible to cover all topics in 8 hours,
Yeah,
sir.
there were
That is
some
the
problems.
problem.
Yeah. चलिए फिर आप बहुत अच्छा लगा आपसे मिलके, और जाते जाते एक और
Thank
शेर
you
सुना
so
के
much,
खत्म
sir.
करते हैं। मैडम एक शेर सुना दूं अगर आपकी इजाजत हो?
हाँ सर,
Yes, sir. Please,
कहता
sir, please.
सितारों को आंखों में महफूज रख लो। महफूज का मतलब होता है उर्दू, मेरे फादर साहब उर्दू के टीचर थे। महफूज का मतलब होता है कि उसको संभाल के रखो। सितारों को आंखों में महफूज रख लो, बहुत दूर तक रात ही रात होगी। मुसाफिर हो तुम भी, मुसाफिर हैं हम भी, कभी ना कभी कहीं ना कहीं फिर आपसे मुलाकात होगी। थैंक यू।
वेटिंग फॉर दैट सर।
Thank you, sir.
Sure.
Thank you, sir.
कभी आएंगे आपके औरंगाबाद में कभी।
Thank you.
जी सर।
Definitely.
Yes, sir. Yes.
गुड, ओके, ऑल द बेस्ट।
Yes, sir.
Thank you very
Thank
much.
you, sir.
Thank
Thank
you.
you, sir.
Thank you very
Thank
much.
you. Thank you, everybody.
so much,
Thank
sir.
you very much.
थैंक यू।
Surve sir, your video once. We didn't see you today, Dr. Surve.
Ma'am, my video is not working. Computer, laptop video is not working. I tried it.
Okay.
But
Because
thank you very
Dr.
much, sir. Thank you very much, everybody. Thank you very much for participation and enthusiastic support. Thank you.
Thank you so much. Okay,
Thank you.
bye.
Goodbye.
Thank you everyone.
Thank you.
Okay, sir. Thanks a lot.
Okay, we will end.
दासे सर आता बोला पुन्हा.
मध्ये डिस्टर्ब झालं थोडंसं.
हरकत नाही सर, जाऊ द्या.
No problem. Very nice.
Congratulations to everyone for successful organization.
Thank you, madam.
Yes.
Congratulations to everybody.
Most awaited
thank you.
workshop. So it is.
Yes.
Congratulations everyone.
चलेगा. चलो, bye.
Thank you. Yes.
Bye,
Bye.
Surve sir.
Thanks a lot to our
Thanks a lot, participants, all participants. Thank you.
Thanks to HOD ma'am.
Thanks to HOD
Yes.
ma'am.
It is all because of your support and your efforts. Swati madam, where is she?
स्वाती मॅडम गायब झाले वाटतं.
चला, bye. Thank you very much.
Hello.
Okay.
Bye. Thank you, madam.
Bye. See you.
Bye.
Mahavir sir, thank you.
Mahavir
is there.
sir is there. Yeah, he is there.
त्यांचा व्हिडिओ ओपन करता येईल का?
Oh,
महाजन
missed Mahavir.
महावीरचा ओपन करता येईल व्हिडिओ? Pallavi, she has left, I think so. Bhavna was there?
महावीर सर बोला.
व्हिडिओ ओपन करा त्यांचं.
व्हिडिओ नाही होणार मॅडम. एक मिनिट बघतो.
नाही का?
बघतो मी.
सुर्वे सर तुम्ही लवकर उठायला लागले.
पॅनलिस्ट केलं तुम्हाला. आता व्हिडिओ आणू ते ऑन करा.
व्हिडिओ ऑन करा.
पुन्हा जॉईन होतील. रिजॉईन करावं लागतं ना त्याच्यामध्ये.
व्हिडिओ होतोय का? व्हिडिओ नाही होते ना?
नाही ते त्यांना पॅनलिस्ट करावं लागतं ना. पॅनलिस्ट केल्यानंतर व्हिडिओ.
दासे पण गेले बघा. दासे कुठे गेले?
आहे मॅडम आहे.
व्हिडिओ तुमचा दिसत.
महावीर सर पुन्हा जॉईन झालं नाही वाटतं.
आले आले आले
आले.
हाव आर यू? व्हिडिओ.
अरे अनम्यूट कर. अनम्यूट.
महावीर.
Ma'am, nicely been hosted and nicely smoothly conducted. So I was so happy being the part of this workshop. Actually मला काही सेशनला लेट झालं actually मॅडम.
तुम्हाला सगळे रेकॉर्डिंग मिळतील.
हो सर थँक्यू सो मच. आणि खूप छान वाटलं सर्वांना बघून पुन्हा एकदा. अँड
हाय सर.
ऍक्च्युली सरांनी शेवटी शेअर चांगले सुनावले. एकदम नाही सर अजून पण घेतले असते पण टाइमिंग होत त्यामुळे वेळेमुळे थोडंसं घेतलं नाही सर. तर तुम्ही व्हिडिओ ऑन करायला पाहिजे होता.
अरे सर ते होत नाहीये. लॅपटॉप ऍक्सेप्ट करत नाहीये. काय माहित.
अरे सुर्वे सर व्हिडिओ बघायचं होतं आम्हाला. व्हिडिओ
ट्राय करतोय अजून. ते नाही म्हणतोय. मेक शुअर दॅट द कॅमेरा इज पॉवर्ड बाय काय इज कनेक्टेड टू युअर कॉम्प्युटर.
ऑर्गनाइज
टी शर्ट घातलाय का म्हणून ते नाही ऍक्सेप्ट
करत.
असं वाटतं
खोटं थोडी बोलणार आहे.
टी शर्ट चेंज करतो.
नाही मॅडम वेल कंडक्टेड मॅडम. ऍक्च्युली क्वेश्चन आन्सर सेशन म्हणजे सगळे को होस्ट वगैरे सगळे व्यवस्थित केलं एक्सप्लेन सगळ्यांनी. कारण क्वेश्चन आन्सर खूप महत्वाचे होते. सरांनी एक्सप्लेन पण केले चांगले. आणि बाकी हाऊ आर यू ऑल बाय द वे?
तुम्ही कसे सांगा. नांदेड काय म्हणते?
हा सगळं व्यवस्थित आहे सर. आमच्या
सर आता
हो नांदेडकर झालो आम्ही. हो तुमची कमी इकडे फक्त सर. We'll meet you soon. We'll meet you after 2 months. My cousin's marriage is there in 30th of October. So I'll be there in Aurangabad.
Okay sir.
Right now we are very busy in COVID. We have been started with our ward duties also. So from next week I will be starting with the ward duties and all that. So hope you will boost my confidence. Yes, of course. You too all take care, sir.
Right, ma'am.
Yes.
HOD ma'am, everyone.
चालेल.
Thank you so much. Thank you so much. बोला.
Thank you sir.
अरे thank you. बोला तुम्ही दिसत नाही हो सर. तुम्ही बोला.
सर ते व्हिडिओ ऑन नाहीये. कोणतं भवन ते तुमचं?
कोण?
तुमच्या नांदेडचं ते क्वारंटाईन सेंटर भवन कोणतं आहे ते?
पंजाब भवन.
बहीण ऍडमिट आहे तिथं.
बर. तुमची सिस्टर आहे का तिकडे?
हा सिस्टर आत्ते बहीण ऍडमिट आहे.
बर बघतो सर बघतो.
हा चालते. चला थँक्यू व्हेरी मच.
ओके थँक्यू ऑल ऑफ यू.
End the meeting now.
Okay.
Yes. Good night. Take care all of you.
Thank you.
Take care all of you.
Yes.
Thank you.